firstly, I would like to introduce him that he is a professor in bioinformatics from School of Computational and Integrative Sciences, Jawaharlal Nehru University. Uh, he has a lot of publication with the citation intake is up to more than 3,000, with the H index is 28. And Professor Shandar will give a talk, very interesting talk on biomedical discovery via data science and deep learning. So without any further ado, Professor Shandar, I would like to invite you for your talk. Uh, 35 minutes, I think, according to the schedule. And time is yours.
Thank you. Thank you, Dr. Hassan, and thank you for inviting me here, the organizers. And these are 35 minutes allocated to me, but if you don't enjoy and if you want me to interrupt before that, you're most welcome to ask me to stop. That is getting too much. Okay, so a very good morning to all of you. I'm speaking from New Delhi, Jawaharlal Nehru University, and it is so nice to connect to Indonesia at this moment. I hope I can receive some of the sun from Indonesia. It is very cold here, and I hope in the virtual world we can exchange some of the um, weather differences, and the day here becomes warmer with your blessings. So with this note, with this quick background word, I start. I will share my screen. Okay. I guess you can see my screen now.
Okay, great.
Okay, so I come from this lab. My lab is called Sci-Y Lab. Those of you who can read a little bit of Japanese can see on this in the star there is a word called Sci-Y. In Japanese, it means happiness. And this is an integration of the scientific Sci and Y is the for query. So my lab is named after stuffing together lots of things. It looks like the scientific query germinates and produces happiness, as in this logo that you see here. Since the time is not so much, I will go to the content. Can you see my slide moving? So I start with a small poem that those of you who may be interested in poetry and also in science, we may enjoy it. Where does the data science stand in comparison to the wet lab science? Because this is a biodiversity conference, and many of you may have never done bioinformatics or never even respected bioinformatics. So I put here a few subjects in contrast in the form of a poem. Physics is fundamentalist because in physics we often talk about fundamental laws of nature. So physics is fundamentalist and fussy. Mathematics is dumb and yet not easy, because mathematics has very well-defined equations, and things cannot be so fuzzy in this unless it is fuzzy mathematics as that has come later. So mathematics is dumb and not yet easy. Astrology is hasty and crazy, which draws conclusions too fast. Human brain uses none. For deep inside its terrain, it knows to represent, test, and train without bothering to explain. And that is what the situation is about intelligence. Intelligence may not specifically identify how and why it is answering a question the way it is answering, but it may still give you very good answers. So the human brain is not based on physics, not based on mathematics. The answers given by human brain, which are intelligent, are not based on any of these basic sciences, but it does represent, it does test hypothesis, and it trains models without bothering to explain how it answered the way it answered. Data science rules this world, for even if a solution is transient, predictions are noisy and incoherent, they work the best for most because they are intelligent. So this is a little primer for, I'm just trying to look directly into this screen. Okay, so I start with, again, since it is a biodiversity conference, I would like to pick up something from more familiar domain from some of you, many of you, and a comparison between the philosophies or approaches to science from Charles Darwin and Gregory Mendel. Both of them were almost contemporary scientists. They both left this world almost within the gap of less than 2 years. And Darwin studied fossil data, and Gregory Mendel did experiments. So there are two great scientists, one involved in experimental and mathematical work, mathematics based on that experiment gave rise to genetics, and the other looking at the fossil data, giving rise to a great evolutionary theory that forms the foundation of a lot of biology today. So now one may wonder, was Darwin a data scientist? Why? Why do I raise this question? Because he collected data. It was a fossil data. He modeled this data to theorize evolution. He explained origin of a species based on data. It was not based on first principles of science. It was based on the observation and phenomenology of the data that he observed. Although he never quantified it, but he definitely created a theory based on the qualitative analysis of data with some quantification coming in the form of dating. So he never went to a lab to create this thesis, or he never performed an experiment for his landmark theory of evolution. So now we wonder if the data science has been as old as in 19th century when Darwin was also doing data science. In fact, all science has been based on some data or the other. Can we call all those science to be data science, or there's something new today which makes data so much more important than before? So that is where the machine learning comes into picture. The machine learning converts the data into models with arbitrary complexity. So
Professor
the whole,
Ahmad, excuse
yes,
me, I interrupted you. Could you
yeah.
close your mic so the voice is louder? So
Oh, voice
some
is
of,
low.
yeah,
Oh, I'm sorry. Did you
okay,
hear all what I said?
yeah, yeah.
Do I have to
Okay, I'm
repeat?
so sorry to interrupt you.
No, no, thank you for interrupting.
Okay,
It's a good interruption.
yeah.
So am I better audible now? It should not be like
Yes,
this.
okay, okay. Yes.
Is it any more helpful? Can you hear me
I
better?
think it's okay. Yeah, fine. Now it's clear.
Okay. So machine learning can convert any data into models with arbitrary complexity. That is, machine learning was not available to Darwin, and machine learning was not available to people who were studying data earlier. So all the science that came from data, Newton's laws of motion, that came from the data on the planetary motion. And the quantum theory came from Bohr's observation of the hydrogen atom spectra, their data again. So these data was modeled carefully using specific mathematical relationships, and then the laws were created. Machine learning was not there at the time, and machine learning was not used to create the fundamental theories from that. So what is new today in the big data analytics, or the data science that we call it sometimes, is that we completely bypass the basic laws. We are not really interested in how my feature set, which are my inputs to the model, and my target properties, say for instance, the transcriptome, the metabolome are my features, and I want to know what are the phenotypes these transcriptomes will produce. So this relationship is not modeled with the help of mathematical equations, but with the help of some black boxes. We do not know how this model is being created, a neural network which has certain hidden nodes, certain input nodes, and certain layers. But what these layers are doing and what is a mathematical relationship it is generating is not generally of our interest, although some efforts are made in that direction. But that's not the primary purpose of training a machine learning model. So this is the fundamental difference between the previous application of data to produce or to make new discovery compared to the discoveries that are being made today from the data science. So I can simply, if I put this data and this modeling or finding rules from data in perspective, we can say that there are three paradigms. In the older paradigm one, the basic statistics and curve fitting was the primary principle. Mathematical modeling were the primary principles of converting data into scientific theories. Then paradigm two came when machine learning and clustering came, and now we are in the paradigm three, where the deep learning and natural language processing and the technologies AI and cloud are there. So this is our way of looking at three paradigms. There are other researchers and scientists, thinkers who have talked about fourth paradigm, etc. But here I put this in the context of how the mathematical modeling has developed over time and how the data is converted to discovery over a period of time. So first, I share with you some of my own experiences, our own research, where we have used these three approaches to solve biomedical problems. Very long ago in 2004, we were looking at the data of DNA binding protein structures, and we wanted to know which of these proteins, if I just know the structure and if I do not know whether this is a DNA binding protein, what are the properties of the proteins I should look at. There were very complex models available at the time. I'll just try to summarize that, and I could identify that if we just count the total number of charged residues and total net charge on the protein, and thereby compute a rough value of a dipole moment and its quadrupole moment using certain approximations from even low resolution structures of proteins, we could predict DNA binding proteins from structures with high degree of accuracy. This was done way back in 2004. And this is where the quality of data comes into play. Here, the model itself is not so sophisticated. Models are simple, but the data that goes into these models are of a very high value or of a very high quality, and then only the simple models can emerge from the data. Otherwise, all data will all data that all it will produce will be noise, and no modeling can be carried out. So that is where these models become successful. This is the older, the traditional, the first paradigm method of looking at the data and making discovery. More recently, in 2014, another high quality data we compiled from Protein Data Bank, and we curated the data of DNA binding proteins undergoing conformational changes. When they bind to DNA, their structure changes. This is very important because if we create a homology model or any low resolution structure, we will probably not know how the protein will actually be when it binds to DNA. And a lot of conformational change takes place in this process. So if we classified these conformational changes into 6 categories, and each of these categories had a distinct signature in terms of the stability and the specificity, for example. And this helped us to make discoveries about these relationships and also predict what will happen to the complex. Will the complex be stable? Will the complex will be weakly stable, and etc. These properties could be related to the conformational change categories that we identified in 2014. So this was the first paradigm, a simple overview of how the simple things can lead to interesting discoveries. Then we come to the second paradigm, because the simple statistical models where we just collect high quality data and we try to model them, we are trying to make some very simplistic assumptions that such as linearity, non-additivity, like x plus y is equal to xy, or there is something that x plus y is more than x plus y. That is, so this is the non-additive behavior. That is, if you, for instance, if you take a practical biological example, it will come, I'll show you in the next slides that if you remove one residue and the energy lost is E1, and if you lose another residue and the energy loss is E2, if I remove both of them, the energy loss is not E1 plus E2. It is either more than E1 plus E2 or less than E1 plus E2. So the residues are interacting with each other. In the context of a computational model, if I want to predict something from a set of features, then these features are interacting with each other. So there is an inherent complexity in my data. So the simple models of statistics, and such as linear regression or simple curve fitting, will not be able to solve these problems. We have to find models which can take cognizance of the complexity of the relationships between the features that we use as the input, for instance, a set of genes, expression values, and the output target properties that we want to see whether a disease is likely to be encoded in this gene expression profile. So this is where the machine learning comes. And we early on, when we were trying to predict the DNA binding residues, whether there's a whole sequence of DNA binding protein, and we want to know which of these residues in the protein sequence actually interact with DNA. So this does not happen in a non-additive way. Every residue participates in interaction with DNA, not on its own, but in cooperation with the surrounding residues. But the surrounding residues do not join or combine with the key residue in a non-additive way. Just again, example, E1 plus E2 is not just an algebraic sum when E1, 2 takes place. So this kind of a non-additive behavior, if we want to introduce it into the simpler model, such as if we want to create a multiple regression model of the interacting pair of features, then the complexity becomes too high. Traditional models, statistical models cannot handle that. So if I want to incorporate a polynomial of where the features can be added as pairs, so the number of parameters that my model requires to train or optimize is n square. So this complexity becomes very, very high. And any model which is based on this high complexity is overfitted and does not have a practical value in the application stage. So that is where the machine learning, particularly neural networks, become very powerful. Neural networks can model very complex surface between the feature space and the target space with very fewer parameters than, say, multiple linear regressions where you have complex features included, or Bayesian models where we want to include the pairs of residues, one dependence models or multiple dependence models. If we want to incorporate the feature interaction into these models, they become very complex. The neural network can model very arbitrary, very difficult, very complex surfaces with much fewer number of parameters, and therefore can avoid overfitting. So that is where the classical machine learning helped, and these are some of the papers we published in this field to predict DNA binding residues with the help of neural networks. From 2004 onwards and 2009 this was the main area of activity for my research. Going further on, we were also looking at the residues, amino acid residues in protein sequences. And as I said, that the different groups of DNA binding proteins, the nature of evolutionary profiles, the conservation of residues was found to be different. And again, we could predict with the help of this, if I can identify a cluster of conserved residue, I can tell this is the cluster of conserved residue which is crucial for binding to DNA or interacting with the genome, and as a transcription factor, initiating the gene expression process. So these two papers we published in Nucleic Acid Research in 2008 and 10, both of them basically talked about this idea of the clusters of conserved residue. The technology here, as you see here, the clustering technology, the machine learning, the unsupervised machine learning, where we do not have a target property, we are just looking at how the charged residues come together and form clusters. And then we just find these modules or these clusters within the protein structure and then characterize them. And then this will be, so in combination of the unsupervised learning in the form of clusters and then subsequent domain analysis of the looking at the biological properties and the thermodynamic data of protein DNA interactions, we were able to identify some very, unravel some very interesting properties and show that they interact in a cooperative manner. So now coming back to the third stage of the current paradigm of pattern mining, I take some time to spend on what is happening today in our lab, particularly in the context of my own research. So first topic, the issue that we are dealing with is the in search of DNA recognition code in cellular contexts. There are many questions about how the transcription factors regulate certain genes. Many questions are still unanswered. For instance, there is a transcription factor, and in the entire genome, there are many occurrences of the same sequence motif. This sequence motif is recognized by this transcription factor in some places, but it is not recognized in some other places. So out of the thousands of genes which have a given target sequence in the promoter region for the same transcription factor, some are regulated in one cell, some are regulated in another cell. So we do not know what causes these particular genes expressing in one cell and some others expressing in another cell, although transcription factor is available in both cases. So this is one primary question. Another question is that the genome is so long, and if the transcription factor has to find where it has to bind, it takes a very long time for transcription factor to scan. But transcription factor can actually quickly find its location where it has to bind. So how does the transcription factor go from completely cellular distinct area like a cellular region and go to the nucleus and then identify where it has to bind? But even more intriguing is that the backbone of the protein The backbone of this DNA is identical everywhere. The bases which determine the sequence of the genome are where are actually not visible to the transcription factor. They are inside the double helix. The double helix backbone is everywhere the same. So how does the transcription factor know from outside what is the base sequence inside this? So this is another question. So there are many questions about the transcription factor binding to their targets, and also protein DNA interactions between in the host pathogen interactions. So these interactions are so complex, it is very difficult to predict computationally, and without actually performing lots of experiments, we hardly get much understanding of what may be happening in this. Some of the questions we have tried to answer using bioinformatics in our lab. So one of them is to do a conformational characterization of DNA, that the DNA double helix is not always a ideal double helix. It undergoes certain deformations. And if you can see in this figure, that those who are not familiar with this can think of these two complementary bases in a double strand, so G and C or A and T. And from the ideal conformation, they are deformed slightly. There may be some shear, there may be some buckle, there may be some stretch. Instead of their ideal double helix, they form this kind of elastically deformed structures. And it is this elastically deformed structures are dependent on the sequence. If it is GC, the value of shear, buckle, stretch will be different. If it is AT, it will be different. If it is GG on one side, CC on another side, it will be different. And if it is GC on one side and CG on the other side, it will be different. So depending on what is the sequence at a given location, the backbone also deforms slightly, and there is what you call elastic excuse me. There is an elastic deformation. And as I said, so since the basis cannot be seen by the transcription factor, what can be seen by transcription factor are these deformations which are visible at the backbone level. So we characterize this, and we developed a method called readout, where we can take protein DNA complex structures from the protein data bank, and we can look at these structures. Okay, this is the sequence, and this is the structure that is required to bind to DNA. Then what are the sequences which can take this structure with ease, with low energy? And what are the other sequences which can take the same? So is the sequence that is placed in protein DNA complex, is the DNA sequence the very highly specific sequence, or many other sequences can form this kind of a structure? So these type of questions we analyzed, and then we created this web server, readout.cyberlab.org, it should be, not ORH, and where people can upload their protein DNA complex data, and they can look at the specificity and stability of a protein DNA complex that will be formed with different sequences. They can thread large number of sequences. They can replace the DNA sequence by their own sequence and see what are the likely changes that will undergo in the protein DNA interaction energies or the specificities. So using this method, of readout, we were able to discover a new non-canonical binding site of STAT3, and this was experimentally confirmed in Osaka University Medical Hospital, where I used to work at, in the neighborhood I used to work there. So this university, they performed the experiment based on our hypothesis, and they were able to identify and characterize this STAT3 binding site in SAA gene. So this work was published again a few years ago in BBA. And this is the whole method. Then we moved on to the next step. Then if we have a protein DNA complex, we can try to find the specificity and new non-canonical sites in the genome. But many a times we do not have a protein DNA complex. So if we do not have a protein DNA complex, can we still model what kind of conformation and dynamics the different regions of the genomes can have? So we developed this, we created a molecular dynamics trajectory for all the tetramers that are possible, and with all some flanking regions possible. So the 12 mers we simulated, and we tried to create a dictionary that if this is, for example, if this is my sequence, this is likely to be the molecular dynamics. So we created a dictionary and the software to predict the molecular dynamics directly from sequence. Instead of doing molecular dynamics in real time, we trained our model with the help of machine learning. We trained them to predict the conformational ensembles directly from sequence without performing the simulations in real time. So that's what done by training the trajectory data that we collected from the collaborators, and that worked quite well. With the help of this, we could explain some very interesting patterns of a STAT3 binding to its target. So I summarize this problem. So am I in the middle, halfway in my time? Can Mr. Professor Hassan, can you just let me try to track my time? Am I halfway?
Yeah, the time is still okay.
I'm halfway, so so just to get my idea where I.
Okay, yes, you still have approximately around 15 minutes.
Okay, so I'm about halfway. Okay, so here is this problem is that STAT3 is a transcription factor, and it recognizes, as I said, the transcription factors recognize different targets, target genes in different cell types. So here it is that the data is collected for STAT3 from RNA, the ChIP-seq experiments for four different cell types. One is the macrophages, the CD4 plus T cells, T20 cells, which are cancer cell lines, and embryonic stem cells. So these four types of cells. And what is very interesting is that the number of sites that are observed in the ChIP-seq experiments, only 35 sites out of several thousand recognition sites for this STAT3, only 35 are recognized in all four cell types. On the other hand, there are large number of sites, 1,547, which are only recognized in macrophages. And on the bottom of this figure, you will see that 2,148 sites are recognized by STAT3 only in ES cells and not in other cells. So this is we wonder, we published these results in 2013 in nucleic acid research. So we wonder what makes the same transcription factor bind to different, recognize different targets and regulate different genes in different cells. And this is further insights into this. If we look at the sites individually, and if we know the canonical motif of a STAT3 is TTC and its complement, half complement GAA, with a spacer of 3 bases usually. So this canonical motif is observed in ESC cells, at embryonic stem cells, only in 28% of the sites which were observed in ChIP-seq experiment. In macrophages, this number is just 8%. In CD4 plus T cells, this number is 20% and so on. So you would see a large number of the transcription factor binding sites do not have the canonical STAT3 motif. And then if we look at create a new motif altogether, they create different types of motifs from the sequences that are collected from ChIP-seq. So before the advent of ChIP-seq technology, this kind of observations were difficult to make. But now this ChIP-seq, 4 different independent studies produced this kind of data. So with the help of this tool that we developed, DynaSeq, and we tried to use machine learning to predict these signatures of different cell types, we were able to identify that it is not just the motif which is important, but the surrounding regions also play a role in the recognition of the transcription factor by the genome. So the target regions are here at the peak, and then information that is required in the surrounding regions dropped systematically, so much so that if we model this with the help of a regression model, we found that there are apparently 4 regions in which the 200 base pair regions surrounding the motif can be divided. In the first region, there is a information drop, and there is a different the correlation between the predicted points of this model and the observed points is 0.57. So the model shows that there are 4 modular regions surrounding the transcription factor binding modes, and then we could interpret them to be, say, the region of this is the main binding region and this is the steering region, so that the transcription factor can quickly go here, and the stabilizing region and the steering region. So 2 regions are the steering regions that help the transcription factor to tell, okay, this is not the place you have to bind, don't spend time here, go quickly to the stabilizing region. And the stabilizing region gives a stable structure for the protein, the transcription factor, to bind its target. So these results we published in 2017, and we were able to then look at not just STAT3, but in overall, but also in different cell types, they have a different profile of the shape. So in CD4 plus T, then the region just surrounding the 2 regions are important. In macrophages, all 4 regions are important. In fact, the actual motif region is less important. So probably the role of cofactors becomes predominant in the case of macrophages. In ES cells, these regions surrounding the motif are roughly continuous, and same is true for cancer cells. And we already know that ES cells and cancer cells should behave more similarly to each other, and the two immune-associated cell types should behave more similar to each other, and that is what also emerged from the analysis of the DNA shape of these sequences surrounding the transcription factor binding motifs. So then going from stat 3, we went to look at the large number of transcription factors, 1,500 transcription factors, and the ChIP-seq data. Their NGS data analysis showed that there are 4 clusters, and they have different types of profiles in terms of the information distribution around the binding motifs. And so there is a group of 229 transcription factors which have a sharp peak where the surrounding regions are not important. Then there are 504 on the other extreme, where the surrounding regions play the predominant role in the interaction with the protein, in the protein DNA interaction of transcription factor. There is another resource that comes from our analysis of the machine learning application to DNA binding proteins, where we try to integrate the gene expression with the sequence information to predict DNA binding proteins. And this became possible. So just I show you this data, that there are the genes that code for DNA binding proteins have typically higher expression value, but this higher expression value is not necessarily be associated with DNA binding activity. This is in combination of these gene ontology terms, we could integrate the gene ontology information about the co-expressed genes and the nature of the co-expression, and we could improve the annotations of DNA binding proteins. Actually, the integration of the expression data and the sequence and the structure data helps us to annotate proteins in the specific context of the expression profiles. That is, in the classical annotations of proteins is like, if this is the sequence and this is the structure, this is the function of the protein. But when this function will actually take place, under which cellular context will this function will be performed, has to be modeled with the help by taking cognizance of the expression signature or the cellular context. So cellular context can be incorporated into the model by the expression profiles. So we try to combine these two and predict the DNA binding function activities with the help of these two features taken together. And we showed that there are some proteins which do not have a very strong sequence level or structure level signature of DNA binding. But if certain genes are co-expressed in a given cell, they may perform this function. So this kind of a mechanism, that the functional compensation which is encoded in the sequence, if the functional encoding in the sequence is not strong enough, that can be compensated by the expression signature of the cell. And this will allow the different types of transcription factors or the proteins to perform different functions in different cell, because the other expression environment also contributes to this same function. Then we applied just a few words about deep learning models. The models that I showed you so far, most of them were the classical machine learning models. In the deep learning model, I have a few words more to say, just to put it in a background, that we have so much of data everywhere, and then we do not have, I just give you the example of dimensionality. Again, I was saying that we are collecting a lot of features for the data, and we often talk about big data, but sometimes we do not have big data. So we have a lot of data sometimes, and we often say in biology there is no data for this. So actually, that there is data required is not what is available. So we have to use the data that we have, and we try to predict and try to model information that is of our interest. So people have been collecting indiscriminate features for the kind of models that we have to create, are such as in MRI images. Even if it is a 600 by 300 pixel image, you have 1,80,000 pixels. This is the dimensionality of my predictive model that I'm trying to create. If I do an ECG diagnostics, these are the problems that we are addressing in our labs. That's why I picked up these problems. So if we are doing a time series analysis of an ECG, and I want to know where this ECG has become erroneous, where is the location of erythemia, then I have again a very high dimensional. If I have a 1 hour recording of for every second, which is the number is, as you can see, 3,600 per hour. And then the thing is, this dimensionality itself may not be a problem, but if we are trying to fit models, and if we have just 10 instances of data that we want to predict to model, and in this case of MRI images also, we do not have more than 100 images. Sometimes we have just 100 images. Like if we want to predict, like we were working on one problem where we wanted to relate the MRI lesions observed in MRI to the linguistic deficiencies. So we had less than 100 images. So the challenge here is same is true for the gene expression data analysis. In all these cases, if you look at co-expression, you have millions of dimensions and just few hundreds of the data instances. So if you have just a few, the number of data instances is fewer than the number of dimensions. The conservative, the classical approach is that the number of data points that you want to fit your model should be more than the dimensionality. But then you see so many models coming for image recognition, image classification, and all. All this, if you are feeding the entire image to the model, you are really feeding a lot of features into the model. And then you are trying to, you do not have enough data to train in the classical context. But then how these models become possible? These models become possible because of something that is called convolution. I don't know if I have that slide here. Yeah, so the convolution will try to first, instead of directly putting into the model, the convolution allows to create a structure within these features. So in some sense, the inherent power of the CNN comes from the fact that the features can be convoluted, and they can actually, the data requirement becomes much less.
You
If
have
we
5 minutes.
5 minutes, okay, yeah.
Yeah, okay.
Okay, thank you. So this the primary challenge for creating these machine learning models or any high dimensionality models is to avoid overfitting so that generalization can be achieved. So this generalization can be achieved with the help of CNN, have been particularly successful in this field because they have an inherent quality of modeling the relationships between these features without actually directly training them. So the convolution layer makes this possible, and then later on they are pooled so that the effective dimensionality can be much less than what it actually, the number of parameters we are training. So this is where we studied the comparison between the shallow, the traditional, and deep learning models for cognitive performances of stroke patients. And that is where we systematically showed what is the data requirement. If we create our model with 50 MRI images, 60 images, 80 images, how the performance goes. And what at the smaller data sizes, classical model worked better. But there is a certain data amount where the deep learning overtakes, and that number can be estimated by a systematic analysis of the modeling the models. And then we used this combination of principal component analysis and the feature based, the CNN, and that this is what I'm showing you, what I was talking about, the convolutional layer, which creates these smaller units, and then it creates a new representation of features. And finally, we use a fully connected layer, which has a much fewer units than we had in the beginning. So we have in the input model 61 into 73 into 61 3D image of MRI, and in the end, actually fully connected model has just 500 units. So this entire modeling from the convolution to the pooling and the fully connected layer is where the actual interaction between pixels is being modeled. And that is what gives power to CNN in contrast to traditional deep model of multi-layer perceptron. So I skipped these results. So this is what we do. We did the comparison. So this is another problem we dealt with the evolutionary profiles of proteins, and we try to predict again their protein functions. And this deep model with the help of CNN performs much better than all the previous models that we have also developed earlier and other researchers have developed. This is ECG I already talked to you about, and here we use the model, the deep model, which is an advanced version of the recurrent neural networks, LSTM, and this combined with the classical models of SVM could predict where exactly the anomaly in an ECG is and what kind of anomaly is in a given ECG signal. So in summary, I summarize this, what I said, data science has always been fundamental to the scientific analysis and discovery. The field has moved from simple statistical modeling to classical machine learning, and now the deep learning models, and also the natural language processing, which I have not touched upon today. So the technologies that are used to this have changed not only in terms of the mathematical and computation model, but also in terms of the infrastructure is created. So now instead of doing this on the mainframes computers, people were doing on mainframe computers earlier, then moved to servers, and now they're moving to clouds. So these from clusters to the public clouds. So this is how the paradigm shift has not just taken place in the form of computational models, but also in terms of the resource planning, and the infrastructure that is used for computing. And a brief discussion I had was about the shallow and deep models. And it is not always that the deep learning model will perform better than the shallow or the traditional classical model. It depends on the model complexity that we want to generate, that the data that we have, really it has that kind of a complexity, and how much data we have. So these have to be taken care of, and we cannot blindly believe that a deep learning model will always do better than shallow or classical machine learning, or even simple statistics. That's the conclusion from today's discussion. And finally, I conclude with this little poetic description of this: mimicking brain, shallow and deep, machines learn when man is asleep. Without complex equations or laws, making use of errors and flaws, that is how the training takes place. Regressive, divisive, and classifying, these are the terms often observed in machine learning. So regressive, divisive, and classifying only in AI are so gratifying. So this is the summary. And finally, people who have contributed to this work, some of my lab students, some of them may not be represented here. Some of them have left. A couple of them have left the lab since, and but I acknowledge them because their work is included here. So thank you. That I stop here.
Okay, thank you very much, Professor Ahmad, and then very interesting and enlightenment talk from your group. And I notice there are some questions on your talk. Let's we continue to the Q&A. Yeah. And I think one question and directly you can answer directly. Okay. Okay. The first question from Umi Baroro from Indonesian School of Pharmacy. Yeah. He would like to ask about the modeling of DNA or RNA. So the question is, how to evaluate our model of DNA or RNA is good. So is there any parameter that can that we can use to scoring our model?
So is this the question, Umi Baro? So I can read it directly from the screen also.
Yeah, yeah, Umi Baroro, yeah.
So hello, my name is Umi, and
how to evaluate.
I'd like to ask about modeling of DNA RNA. So in general, I'll say give a general answer so it can help other researchers also. How we so the question is how we evaluate whether my model is working or not, right?
Yeah.
So there are this is a fundamental question of machine learning, because my model may be working very well on the data I am training. That is not what we want. We don't want our models to memorize. We want our models to learn. And there is a difference between memorizing and learning. So the key question of machine learning models is to ensure that the model works on the data the model has not seen. So there are two validation. There are two types of validation. One is the technical validation of your model, and the other is the biological validation of the model. So in the technical validation of the model, the primary strategies used are the cross validation. So you do not train your model, or you do not create your model from the entire data that you have. You do everything on your data, training, testing, everything on part of the data. And you leave some data out. Never use this data more than once. Only after you have completed all your training, then use this data to test what is the performance. So this kind of a strategy should be used. But here people do use this strategy, but what they do is they keep on training the model, then again test on this left out data, then okay, say this model is not working, go to another model, and then test again. So this is a wrong strategy that people often adopt, and therefore they can end up overfitting their model and trying to exaggerate the performance, predictive performance of their model. The ideal way is complete your model, train your model, test your accuracy on some data, and then in the end, use your data which you did not use for the training to test and validate your model. This is the primary method. This is the technical validation. And the biological validation is after your model has generated certain hypothesis, you do a domain analysis, look at the literature. Is there additional evidence for the hypothesis that it has created? If because many a times the computationally predicted model is not the right model. Biology is more complex than your model can capture. In your model, all the determinants that determine the feature, that the properties that you are predicting, every property is not incorporated. Every factor that contributes to that hypothesis has not been included. Therefore, there is a strong chance that your model is producing a completely unreasonable hypothesis or an impossible hypothesis. From a computational point of view, it is very feasible and it is very accurate. But from the biological scientific point of view, this is absurd. So this biological validation has to go hand in hand, and that is how we can avoid um, reaching wrong hypothesis.
Thank you. Uh, Second question about from Heru. The question is, there are many models that exist in the machine learning. What model is used in this research? How did you choose the method, and what is the level of accuracy?
Yeah. So here I briefly actually did touch upon this. So, but for a quick guide, what one should do is one should always take the minimalist approach. A good model is not the one which gives you high accuracy on the training data. The good model is which will perform well on the data it has not seen. So a minimalist approach is that if you can get good accuracy by smaller, simpler models with fewer features, then it is accurate. If you are going to more complex models, then these complex models will have by default, and they are guaranteed to give you higher accuracies because you are training more parameters. So there are strategies to penalize model accuracies for the number of features they are using. But in general, if your data does not require, say, a very complex CNN, do not use CNN. Go for a simple Bayesian model or a linear regression model. But if you have lots of data, and the linear regression model will not be able, because you know that this model, this property that you're trying to predict cannot be predicted, is not a linear combination of my features, then you have to go for more complex models. So systematically increasing the complexity of the model is an ideal way. Do not go to the very complex model from the beginning. Start modeling it from a small, less complex model, and see how your performance of the model improves as you make it more complex, as you introduce more features, or as you introduce more parameters in your prediction model. That's a quick guide, but of course, a lot of factors contribute to the model. Sometimes we already know that this type of model has been applied in similar problems, then we can directly utilize that. And because these researchers may have already done the exercise of going from simpler to higher models. So this is what has to be taken care of.
Okay, thank you for the answers. Another question is about the number of the data. Yeah, number of for data training in molecular machine learning model. So what is the minimum number, and what if there are so many outliers that cannot be fit or generalized? From Huyop, University of Yarsi.
Yes. So in it, a rule of thumb in the as I said, in a classical context, the rule of thumb is that the number of data points should be more than the number of features that you are trying, or the number of parameters you are training. But that does not hold anymore in the case of deep learning. With the parameters we train are usually many more than the number of data objects that you have. But this becomes possible because there is an effectively dimensional reduction inherent into these models. So typically, the data size, we can actually model how in our paper that I showed you in MRI cases, we tried to create different models, and we plotted how the performance of the model is increasing um, with the increase in data size. So instead of training the entire data, we select a smaller data, we check the performance, and we keep on systematically doing this. And we keep on comparing how the classical machine learning SVM is doing and compared to CNN is doing. And we find that a threshold, that at some extrapolating this curve, we could say, okay, if I had these many images, if I have this much data, my deep learning will perform better. So this can be systematically investigated, but there is no clear answer into the number of data points. One very important thing that one should remember when discussing this is that it is not just the number of data points which is important. It is also about how diverse are my data examples. If I have many occurrences of the same type of data, then it is not really a lot of data. You may have, like as you say, that if a book has the same word written all over a million times, it does not make it a big book. You could have just you could have written that word into the number of times it occurs. So the amount of information, amount of data is not important alone. The amount of information is important, and amount of information means that you should have sufficient diversity and sufficiently representative examples in your data that you are using for training. So like if you want to create a model for say Indonesian population about some disease, so these samples that you collect should not just come from one hospital or one locality, because if you do this, your models will be biased for only that particular part. So your data has to be sufficiently diverse. So these are the sampling strategies that people have already always studied. So but this is very much important for machine learning also, that your data has to be diverse. It has to have the sufficient variance in the form at each feature level and as a collection.
Okay, thank you, Professor Ahmad. And I think the last question, yeah, due to the limitation of the time, the question from Dr. Ahmad from a student of computer science in IPB University. His question is, could you explain the main difference between using statistical approach, machine learning, and deep learning? In what case we should use the approach properly? So, thank you.
Main difference between using yeah. So uh, there is always a debate what is better, statistical approach or machine learning approach. So a statistical approach is based on usually some kind of a predefined model. Whereas deep learning or the machine learning approaches, when we talk of deep learning, usually we are talking some variant of a neural network. So one challenge in the statistical models is that if as I briefly touched upon during my presentation, that if you want to introduce the cooperation between features, cooperativity between features. Suppose my model has 10 features and my outcome depends on these 10 features. And if these 10 features are independent completely of one another, I can very well use a Bayesian model. A statistical model should work very well. Of course, there will be problems into cross-validation and all. So these ideas of cross-validation come from actually the machine learning regime. But then there are in statistics, you also have the statistical significance, and you can take care of those things, regularization strategies, as second part of your question is also referring to. So these statistical models are here assuming in a naive Bayesian classifier, they assume all the features, all these 10 features are acting independently. But if there is a relationship between these features, feature 1 or feature 2, or feature 1 into feature 2, or feature 1 but not feature 2, if this is the relationship that is important for determining my outcome, this cannot be modeled using simple naive Bayesian classifier. So to introduce this kind of a relationship in a statistical model, what you need to do is to have new features engineered from a combination of these features. So typically you may have features F1 to F10, and then you may have feature 1 into F2, feature 1 into feature F3, etc. So the moment you go to the second level of complexity, you see the number of engineered features becomes huge. And the number of, if you are optimizing it using statistical principles, the number of parameters becomes too large, and you will not have so much of data to model such a complex relationship. So like you have simple models, even the simplest models like one dependence models are also high dimensional models. On the other hand, if you use a neural network which has all the 10 features as the input and just 2 nodes in the hidden layer, you are not increasing the number of parameters in an exponential manner. You are increasing the number of parameters only slightly. And the kind of surface, the decision surface that this slightly complex model can be as complex as the Bayesian model with one dependence or two dependence or multiple dependencies that can be created. So obviously, in terms of this, in this comparison, the machine learning has an advantage. So you have to look at how much data you have, how many features you have, and what is the expected relationship between features. And then you can make a choice whether a statistical analysis, a statistical model is more appropriate for your data, or machine learning or deep learning are more appropriate. So your second question is why used ridge and not lasso. In fact, we use elastic net, which is a combination of these features. So there is a whole discussion when regularization works better than the other. It depends on whether your features are too far from the mean, or how many, if the problem is of having too many features in the model. So this is a larger discussion.
Okay, thank you very much, Professor Ahmad. I think we have approached the time, and this is the end time of the keynote talk for the first. And again, I would like to thank for your time. I think in India now is still very early morning, because some years ago I was in.
So I think
I was in JNU. Yeah.
Yeah, Indonesian friends
Yeah,
have
I was in
taken
JNU
responsibility.
Yeah.
Okay.
2 years ago, I was in JNU.
Oh,
And
nice.
then thank you very much for your kindness. And then everyone who would like to talk and to discuss more deeply about the research and the possibility of collaboration, I think we can go to you can send the email to Professor Ahmad directly, I guess. And before we ended the first session, there is a message from administration. We have to take a photo together. And then, okay, I think from the IT, please.
All right, everyone, I will help with the photo taking. I will wait for everyone to turn on their camera. Please get your camera ready. We'll take a fun photo session together. Hang on. I'm still gonna wait for a few more seconds. All right, I'm gonna take the first one. Get ready. 1, 2, 3. Okay, the second round, because we have 6 room here. 6 screen, 2, 3. Okay, next the third one. And then the fourth one. 2 more because we have 6 screens. Maybe someone with a bigger screen could help taking. And then the last one. This is very fun because we can see the result immediately. All right, done. Thank you everyone for your help. I return to the MC. Sorry, the moderator.
Okay. Thank you, Professor Ahmad.
Thank you
for your kindness. Then we will continue to the second keynote talk from Professor Fahrur. Professor Fahrur Zaman Huyop. Yeah. He's a professor in microbiology and biochemistry, and especially on pollutant degradation. It was very nice talk, nice field, because I'm from the same background actually. And he's a professor from University, excuse me,
technology.
University Technology Malaysia,
Yeah.
in Johor, in Malaysia. And he graduated from UK for BSc and PhD. And then his field of specialization is mostly microbiology, biochemistry, molecular biology of pollutant degradation. So without any further ado, I would like to invite Professor Fahrur Huyop for his presentation. So time is yours.
Thank you very much for brief introduction just now. First of all, I would like to say thanks to the chairman of the conference, Dr. Vishnu Ananta, for inviting me to be here this morning, and also to the moderator this morning, Dr. Hassan, for brief introduction of my previous experience. I'm actually not expert in the field of bioinformatics myself, but I'm using, I apply bioinformatics to achieve what you call it, whatever data that we have in our lab. So that's the study that I wanted to share this morning. So it's actually what is modeling. We use once we cannot access the protein itself, when there is some limitation in terms of to structure or to model or to make the protein crystal. So that's why we turn into modeling. Okay. So because we find some limitation in some studies, not all protein can be crystallized. So that when we cannot crystal the proteins, therefore we cannot really actually understand the whole structure of the proteins and how this protein will interact with the substrates. So that's why at the end of the day, since we cannot model the protein, we cannot access the proteins, therefore we go for modeling. So I enjoy Professor Ahmad just now related to this, to the talk that he told us. So before we model things, it is to make sure that how useful, how confident our model with the previous studies. So therefore, it's actually we run several tests. We use established model. People do experiment, and we do it in model system. So therefore, we can at least estimate how reliable our data based on the the known data that we analyze. So this is something that a logical ideas from there. As you can see, there are many proteins you can model. What I'm interested in, something like proteins that can degrade pollutants. There are many kinds of pollutant. Nowadays, it's very popular in terms of microplastic degradation. But as for me, we look at aliphatic acid. So any enzymes that can react with aliphatic acid structure is very simple structure. So that there will be my interest of my investigation related to the protein-like studies. So not only you study about the chemicals released into the system, the chemicals can come from from pesticide, herbicides, and then in the sea, ocean. So we have solid waste. It's a problem as well. But of course, the solid waste cannot be degraded directly. So it takes time for this. Sometimes you just burn it up. We collect it and then burn this kind of solid waste. And then what happened to our corals? So due to pollution, the corals turn to something like brownish, things like that. So this is the system that we observe daily in news and so on. So not only by this risk of chemicals, so you can see that the oil spill also can give problems. Drilling, because we consume a lot of petroleum substance for motorcycle, car, and so on. So these activities also can cause, if it happen, something like oil spill can also a disaster to the animals mostly. So marine ecosystem is very fragile because marine pollution poses threat to our food quality and human health. So pollution of the oceans can have serious impact in many localities and the regions. So this is why I'm very interested my investigation to seek a new enzyme, a new protective enzyme that can degrade pollution. So we focus on halogenated compound. So most, at least in this chemical, you have one halogen atom. So constitute large group of environmental chemicals. So this is what we always find in industry and of course in some agricultural products. So what we wanted to have a look in pollutant degradation is environment release of these halogen compounds and the resulted in research into biodegradation. So this is what most of the people wanted to study about. the properties, the bacteria characterization, the protein characterization, and so on. Some of these compounds are degraded under aerobic and anaerobic. Some, of course, due to recalcitrance. So these chemicals cannot be degraded at all in our system. So it depends. So how this bacteria can actually produce dehalogenases? This is good question, because this bacteria already exposed to the naturally occurring environment. So you have some plants, some marine organisms, some seaweeds. So it does produce this, what you call it, halogenated compound. So that's where once the organism already exposed at early stage of their, so that's why this organism can develop into a system whereby they produce a specialized enzyme, we call it dehalogenase enzyme, that can degrade all these chemicals. So that's how we believe through means of evolution from many years, organism exposed to this naturally occurring, and now once we introduce any chemicals, the organism, some of the bacteria, they possess all these special enzyme. But for E. coli, of course, you cannot find any dehalogenase from E. coli, because E. coli never exposed. Maybe it's never exposed. We don't know. It always stays in our stomach, for example. But for naturally occurring substance, the organism that exposed naturally, so that's where maybe it has some kind of enzyme gene to produce this kind of dehalogenase. So when we look at this complex cycles in our system, so where does this these chemicals can react? So why we call it toxic, these chemicals? Because some of these chemicals can react with a specific pathways. Something like fluoroacetate, for example, it can react with aconitase enzyme. So once this fluoroacetate react with aconitase enzyme, so it will block the whole TCA cycle. So that's why this halogenated compound is very dangerous. For instance, it can interfere with some of the biochemical pathways. So let us consider very simple herbicide structure. So you have 3 carbons, and normally the halogenase enzyme can react with alpha, carbon alpha. So you have halides, chloride, for example, attached to carbon alpha. So by means dehalogenase can always cleave carbon halogen. So this dehalogenase can react with the first carbon halogen, and the second carbon halogen also dehalogenase can react with. So both halides will be released. So that's the function of dehalogenase enzyme that I wanted to share with you regarding how to model the proteins. Then you have from 2,2-dichloro. So you have also the isomer, both whether it has at D position or L position, for example, DL2-chloropropane acid. So you have this halide whether at D position of L position. This is racemic mixture. So if you purchase from the companies, so maybe you have 50% D position and 50% L position, 50-50. So we call it DL2-chloropropane acid. So some of these dehalogenase, apart from they can react with L position, some dehalogenase can react with the D position of carbon halogen. So this is the beauty of dehalogenase. They are specific. It shows the specificity, and some of dehalogenase, they are non-stereospecific. That means it can react with both D and L position of this carbon halogen. And then from this diagram also, I mean the product of dehalogenation, pervic acid is the product dehalogenation from 2,2-dichloro, and lactic acid is the product of the dehalogenation of D or L chloropropane acid. So this is something like the function of dehalogenase. So apart from, we understand from alpha haloalkanoic, so you have a beta haloalkanoic. So what does means beta? Beta means a position of halides at carbon number 3. So for example, you have 3-chloro. So it is very rare you can find dehalogenase can react with beta position. But for alpha, carbon alpha is very common, especially L dehalogenase. L specific is very common in the environment. So that's why we use this model to make the structure and function, because it is a common enzyme. So it is very easy for us to predict, since we cannot access the protein itself. So that's the rationale of the project that we end up at the moment. So we start from rhizobium. Of course, you know, rhizobium, the structure is like a rod in shape. It is rod in shape, and then it can grow well in 2,2-dichloropropylene acid. So 2,2-dichloro is a single carbon that this bacteria can grow on. It is like dehalogenase, like an inducible enzyme. So this bacteria rhizobium, once it grows in glucose, there will be no expression of dehalogenase. But once these bacteria grow in 2,2-dichloro or DL2-chloro, this bacteria can produce dehalogenase. So it is an inducible enzyme, because what we believe that these chemicals can enter the cells, and then by means of expression, so it will induce the gene in the system. So that's what we call it dehalogenase and inducible enzyme. Apart from, we can always group the enzyme based on their specificity. So for example, group 1, group 2, group 1 shows that this dehalogenase can act on both D and L. That shows non-stereo. Whereas group 2 is specific. Dehalogenase can react with L isomer only, especially at alpha haloalkylonic. So this is very common compared to group 1. So that's why we use this as our model to characterize for this kind of proteins. And then of course we have group 3. It is a different mechanism. So the enzyme can react with group 3, for example, 3-chloropropylene acid. But it is very rare. You can find in a bacteria that can do this job. So essentially, we wanted to illustrate the mechanism of dehalogenase L, we call it, and enhance the dehalogenation ability and help to address the problem environmental pollution. So that's, I mean, that's the common strategy that why we wanted to do this. So many bacteria produce dehalogenase L, and then very few they solve the 3D structure. So some people may declare that they go to the space to zero gravity. Why? Because they claim that at zero gravity you can easily crystallize the protein. But in our case, we cannot go to the outer space to do it. So some scientists, they have a lot of finance implications, so they can always travel to space, work in space just to get protein model. But it is not the case for our group. So far for L2 L type dehalogenase, there are 3 bacteria that already established in terms of their model. They already crystal the proteins. So we use this to mimic our protein. So the 3D structure of rhizobium DHL is neither never been doing. So that's why we wanted to achieve what we call it the protein modeling studies. So that's the aim. To analyze dehalogenation. We wanted to study the mechanistic, the system, and to predict the structure, and then to see what are the catalytic important acid amino involved. So the 3D to predict. So we wanted to predict the 3D structure, and then to model the DHL substrate binding form conformation, and then to define the active amino acids present, and then of course we want to identify the important catalytic amino acid residues based on the structure that we wanted to model. So this is typical techniques that we apply in our laboratory. So of course we have our own acid amino sequence that we already deposit in the NCBI bank. And then we started to structural modeling. So using several, mechanism, computer programming that I'm sometimes not familiar with so many computing software available. And then we assess the structures by means the secondary structure, and then you do the refinement by means of MD simulation. So it is very important to study this, to see how stable the in terms of the dynamics of the atoms and so on. And then we do some evaluation studies to look at how reliable the structure. And then finally we look at the active site prediction and refinement, and then we do the docking analysis to see how good the ligands will interact with this catalytic with the substructure. And then of course this is the end of the studies that we wanted to see how what are the important acid amino. So from this kind of studies, maybe we can doing something like the experimental work. Once we do the modeling, and then we do cytotoxic mutagenesis using modeling, and finally we go to the bench. So we can limit our time frame from trial and error in the lab because it's very expensive. All this molecular work is very expensive. But when you do this kind of modeling, it's very fast. You can always predict the outcome from your data analysis. So since when we do sequence comparison, when we blast or when we do sequence alignment, so very limited. It has very low identity with the non-dehydrogenase from the same group. So that's why we call it, we wanted to do something like protein threading, when there is no significant homology found with our proteins. So we have to look at several options. So we look at many proteins available in the database, and then from this, some of the proteins that already established their model. So we will use this model to model our proteins known as DHL. So number one is our rhizobium RC1 and dehydrogenase type L. That's the accession number, and this is the study of our of the current investigation. Of course, we will do something like multi alignments. So most of these identity acid amino located at the end terminal regions. So the first 50 acid amino, so you can see that this most of the red in color, and the rest become lesser and lesser. And then number two, of course, you can see the sizes of the proteins, full primary acid amino, some are longer, some are shorter. So that's why sequence comparison become complicated. So we can also using phylogenetic tree to see our proteins, how is it related to other proteins. So of course you can see that they are very far. I mean they are very less than 20% identity to the whole proteins that we have from the database. So from that, there's one proteins that are, once we do the alignments, the pairwise, it has some, because LDEX proteins already established, it's a specialized acid amino that can use in catalysis. So once we do this identity pair up, so we can see that we can predict that, like aspartate and then arginine and the second aspartate and serine, they are very closely related between their acid amino and our acid amino. But of course, you cannot get 100% identity like aspartate 1310. So we predict that that's maybe it, maybe the one that important in nucleophilic attack. So arginine 51 with 41, also we can always say that, well, this is a 10 gap that closer to our the location. So maybe that's the protein that important in terms of the catalysis. From that, so we also double check with multiple sequence alignment to see how DHL related to other crystallized proteins. So you can see that some of them, like D position aspartate number 10, is mostly identical to all acid amino that you see here. And apart from others, you have maybe three, maybe three. So most of them 3. So that's some numbers that can give us some confidence that maybe these are the acid amino involved in terms of the binding of the substrate to the proteins. So this is like you play like jigsaw puzzles. So we just looking at the type of acid amino, and number 2, you are looking at what are the position of the acid amino. Of course, the difference in terms of numbers, the location, because due to the fact that the length, they are not accurately can be measured. So some longer and some shorter. So that's why you can see varieties in terms of the number, location of acid amino. So I put red in color. These are the important acid amino that we predict based on the ALDEX structure. So something like aspartate D, arginine 44, serine, and aspartate 207. These are our early prediction before we model it. Of course, the protein is very sensitive. Whenever you have some substrate with protein, so the protein started to change the conformational. So this is very dynamic. I mean, the software that we use is very dynamic. So initial characterization, our acid amino consists of 279 acid amino, but maybe other proteins maybe less or maybe more. And then we can always predict in terms of protein parameter. You can see that the molecular weight, how much, and then you predict the PI numbers. But of course, PI you can always check the PI numbers, and then by looking at the number of acid amino, you can say how much it is negative. 31 and 34 is positive charge residues in terms of acid amino. So you can always do that theoretically. And then you wanted to characterize based on the hydrophobicity also, you can do it. I mean hydrophobicity test to get the gravity values. So in our case, we have a positive values, which is generally we can say that it is most hydrophobic. So this information must tally to each other in terms of acid amino numbers and so on. So the tertiary structure then sequence the identity between the L and soft PDB template. So that's why we do. We use several target templates to do the alignments. We call it threading methods to demonstrate to produce more accurate model than sequence alignment comparative modeling approach. So that's what we are doing at the moment. Okay, threading. Yeah, I mean, yeah. So when we do the refinement evaluation homology model, we use MMD, molecular dynamic simulation, to see how the proteins, how stable the proteins within a specific time frame. So we choose that maybe 10 nanosecond. People say that maybe 20 and so on. So in our case, it is stable up to the 10 nanosecond to do this refinement analysis. And then, of course, you have to check with several tools, whether to check the stereochemistry, the fitness, and non-bonded interactions, and so on. So we set the cutoff points is 50%. So anything above 50%, so we can always rely on this data. And then, of course, using secondary structural modeling, you can always see the alpha helix, beta sheets. So you can see that alpha 1, 2, 3, 4 located at the subdomain. And of course, you have beta sheet. Beta sheet 1, 2, 3, 4, 5. There are six. There are six beta sheets structure present located. Some of them between B4, B1, B2, B3 at main domain, and the rest at the C terminal regions. So this is how we can see very clearly in terms of the secondary structure model. Then of course we get the refined DHL 3D structure, so you can see that the alpha helix and then the beta sheet as well in the protein structure. You can, using the software, you can always 3D software, you can see north and south, left and right, and so on. But I'm not really expert with the software itself. So this is we just take the 1D photos to see what are the appropriate structure, and we can figure out what to do next in the system. Then we, of course, you need to superposition with the one that already crystallized with ALDEX. We compare that with the shell. It looks like more or less they fall in the same positions between the shell and the ALDEX. So then once we do, then we wanted to study the active site definition and refinement. So using MD simulation, then we do the complex. You can do empty and can always complex the proteins with the substrates. So here we use L2CP, L2 chlorophoric acid as a specific substrate to bind with the DHL, and then resulted in something like conformation change of the protein itself. So the acid amino residues surrounding started to change. So the important acid amino that you can see, something like aspartate, glycine, 16, threonine, they are similar, some are totally different from what we predicted earlier. So based on this, DHL in the study compared to LDEX, this is the early studies that you can see the aspartate still maintained, and then threonine, arginine, whereas when you see that the serine is not the same as what after we do the complex, the modeling, you have a different acid amino that are present throughout until you have some others. So this is something that the prediction that we don't know how true this prediction, but we still rely this data if we wanted to do some lab work, to do some mutation studies and so on. But before we go to wet lab, we can always do mutation studies using bioinformatic system. Then we see the modeling dynamic simulation profiles. So the dynamic stability of DHL and L2C complex and that of the non-complex DHL, we evaluated them, and DHL active site that's occupied with L2C is relatively stable. So you just look at the early structure, and then of course when you see that up to 30 seconds, it looks like they are much more stable. So we assume that they are more or less we can rely this kind of profiles. Of course, then we do the simulation to see how well the L2CP complex with this with the catalytic site or these important acid amino that involve. So you see that the aspartate 13, glycine, threonine, proline, methionine, arginine, histidine, and alanine and isoleucine. So they are well located in this pocket size. And then of course you have L2CP there. The L2CP, the chlorine, that's the chlorine carbon halide, is green in color, and then white in color is hydrogen bonding oxygen atom, red in color is oxygen. Then all the brown receptor structure is represented by the brown, and of course you can see that hydrogen bonding is the thin blue lines. So that's how, and then of course hydrogen bonds, thin blue lines, and the dark dotted line represent the distance between the atom. So as you can see that the aspartate, the location of aspartate 4 angstrom size, I mean that's most probably the one that can cause the nucleophilic attacks to the carbon halogen binding. So that's what we measure the distance. I mean, we believe that the closer they are, I mean that's the most related compared to other acid amino that are present. Then hydrogen bonding between L2CP is the ligand, hydroxyl hydrogen atoms of the carboxylate group, threonine 17. And then the distance aspartate with the carbon alpha that I said just now at 4 angstrom that we measure. And then aspartate, carbon alpha and chlorine atom is about, I mean, in terms of the angle is 115 angles measured by the system. So that's where we believe that the nucleophilic attack started with. And then other acid amino important as well that trap in the cradle forms like arginine 51 and then histidine 184 and alanine isoleucine 186. That's the important acid amino, maybe to bind to make the substrate stable. And then the type of cradle was not found in non-complex DHL. So and then arginine 51. Rotated about 90 degrees toward the center of the active site in absence of L2CP. I mean, that's the theoretical values that can always derive from the system.
Professor Fahrur, you have 5 minutes left.
Yeah, thank you. So since this is hydrolytic enzymes, we look at the water molecules, WT1, WT2, that's the water molecules also located at this catalytic sites. That's the important to make sure that the enzyme react in the presence of water. We call it catalytic water molecules. Based on the theory, you can see that the enzyme, that's the enzyme, and then react with the L-halo acid. So release of chlorides, then you have ester intermediates, and then you have D-hydroxy because chloride release and replaced by OH. So that's why we have D2 hydroxyalkanoic. So this is because the special of this enzyme, you have L become D-hydroxyalkanoic. This is something like that we don't understand in terms of the position of the protein itself. Based on these studies, so we propose the Deschelles reaction mechanism. So we know that the importance of histidine and the importance of aspartate 13 in the system. So you have aspartate 13 is the one that can cause the nucleophilic attack to the carbon halogen of the L-halo acid, and then release the chlorides ions. And then you have the histidine 1 and 4 in terms of hydrolysis. So the histidine 1 and 4 will react with the water molecules. That's what the importance of water that we have, WT1, WT2 at the catalytic sites. So that's the nearest. So H2O will react with the aspartate. So the aspartate is actually can cause the intermediate to release the enzyme and the enzyme and the substrate now become a product. After release of product become a product. So released to get the products. So you have the hydroxyalkanoic. So after this kind of reactions. So that's the importance of acid amino that we predict that DA13 and histidine 1 and 4 is the most important. So how to check this? You can always do modeling studies to replace this acid amino with different other acid amino, see what's happening in the system. There will be another investigation has been done by the other PhD students. So the mechanism of enzyme, so this is the original model that developed of important acid amino involved, like this is not our model, but we took it from LDEX YL, established in terms of the acid amino present, arginine 41, and aspartate number 10, similar with aspartate 13, for example. So, and then you have serine and other arginine is the one that something like to do the hydrolysis of the ester intermediates. That's equivalent to our histidine that we have in our investigation. So, of course, you can see sometimes the acid amino might not similar because due to the fact that once we do the modeling, it changes the conformation structure of the proteins. Then, of course, some of the proteins is not what as we predicted earlier. So this is us, because we are microbiologists, we are so happy to get this kind of observation that we hope that we can study the enzymes. We can do some mutation studies. So to see how reliable this observation. So future work should consider something like to check the mutations and identify the hotspots, and then optimize by combination mutation and so on before we can do the lab work. I think that's all that I wanted to share related to this bioinformatics conference. So I'm very happy to meet with colleagues, not from Malaysia, from all over the world, from India for the first time. To interact with international people, but Indonesia, I always, because it's very close to Malaysia. So sometimes before COVID-19, 2 or 3 times I traveled to Indonesia. So, but now due to the COVID-19, we always communicate through the online system. So thank you so much, Dr. Hassan, for allowing me to deliver my my talk, to share my experience related to very basic modeling studies of protein degradation enzyme. Thank you very much.
Thank you very much, Professor Fahrur, and it was very enlightenment talk. It was remind me about my PhD studies on haloalkane halogenesis actually. So it was very interesting talk, and there are some question on your talk. It was, I think, a lot. Okay, I think we can directly to the question, Professor Fahrur. Yeah. First question from Annie. This is from Industrial Charun Pokpal Group. She has a question on about the application. Yeah. So your research result can apply for bioremediation in land-based because pesticide or plastic residues exposed. How long about the treatment? I mean, I think it's related to if the enzyme is implemented in the industrial. So that's the answers.
Yeah. We cannot predict how long the reaction can take place, because you must remember in pollutant degradation, you must make sure that number one, most based on our experience, it is something like we call it inducible enzyme. Inducible enzymes means we must make sure that the substrate can enter the bacterial cells. So sometimes the substrate cannot enter the bacterial cells, so that makes it is almost impossible to release the proteins. So you must understand in terms of is it possible or not possible for the substrate to enter the cells? So there must be a mechanism of uptake system to allow the substrate enter the cells. Once the substrate enter the cells, what happens next is the gene expression. So the substrate must interact with some other proteins, for example, before it can bind to promoter operator regions, before the expression can happen. So there's other steps. So it depends. Sometimes the substrate cannot even, induce because it is not, in terms of mechanism uptake, it's not present. So there will be no treatment at all for this. So sometimes you can see the plastic or whatever chemicals is just not being degraded, not being utilized by most of the bacteria because this condition, or maybe too toxic. If it's too toxic, the chemicals too toxic, no bacteria can grow. So that will hinder the process of biodegradation as well. So these are many factors that we need to consider.
Okay.
Before degradation can happen.
Thank you. Okay, continue to the second question from Department Biology, Diponegoro University. He would like to ask about, can dehalogenated enzyme apply to medical approach? And what the most important characteristic aspect of dehalogenated enzyme?
I'm not really sure actually whether it can do in terms of medical use. So it is this kind. If you in Africa, there is a plant, Chapitellum, in Africa only present. It has fluoroacetate. So fluoroacetate is very toxic. So the cows consume this plant will die. So so this is very toxic. So I'm not sure how the treatment all about. So of course, but for human beings so far, I'm not sure whether we will be killed when we consume this plant species. But for cows, yes, it is very dangerous. But I'm not sure really the answer for that. I'm very sorry.
Okay. There's another still some time. Probably I would like to ask in the chat. There is no question from my side, Professor Fahrur. I would like to ask about the mechanism of your enzyme actually. According to my experience, that the type of dehalogenases use the halide stabilizing residues, to like capturing the to bind the halogen ions, In the case of the halo acid, is there also use the similar pattern of the mechanism as haloalkane dehalogenases? Because I noticed from the reaction mechanism, you didn't mention about this particular amino acid which act as stabilizing residues of the halide.
Yes, I agree with you. So in some literature, it mentioned that some of these acid amino they are important in terms of to stabilize. The location of the ligands. Yes, I agree with you in that sense. But in my studies at the moment, we look at a very important acid amino that can cause, number one, the nucleophilic attack. So that's number one. So number two, we wanted to check what are the acid amino that can activate the water molecules. So the water molecules will interact with this
Exactly. Okay, I think this is the end of our of the time. So thank you very much again for Professor Fahrur. Probably everybody who would like to collaborate or discussion about your work on, especially on the halogenating enzyme, right? You can contact directly via email. Probably this is the best way to contact Professor Fahrur for further discussion and collaboration. And I get message from the from the IT. We have another photo session again, Professor Fahrur. Professor Shandar, you still there? I think I can see. I hope you also enjoyed
it. Shortly. Thank you for.
Okay, so I give to the IT, please.
Okay, we'll wait for photo sessions. So please, for all the participants, open your video. For Mr. Fahrur, maybe. Okay, I will count. 1, 2, 3. Okay, for the second screen, please prepare. And open your video. Okay, I will count 1, 2, 3. Okay, the third one. Okay, 1, 2, 3. Okay, thank you very much. I will give back to moderator, please, Mr. Ahmad.
Okay, thank you very much for to the IT team. We continue to the keynote number 3, and I would like to invite Mrs. Yu Yin Tan. Yeah, he's an executive bioinformatics specialist in Asia Pacific, Japan from Illumina. Please, Professor Mrs. Tan, are you there? Please unmute your speaker, please. Yeah.
Hi everyone.
Uh-huh.
YY here. Can you hear me?
Okay. And Mrs. Tan has a 10 years experience on bioinformatics, and so since 2012 she joined Illumina, and she had a tremendous achievement supporting key user enables projects, so showing value of her work through its outcome. So also she's passionate about driving customer success through Illumina informatics solution. Prior to joining Illumina, Mrs. Tan was also part of the comparative genomic group at the Institute of Molecular and Cell Biology, A-Star, Singapore, where she dedicated her work to cartilaginous vertebrate genomes. So her current focus is on driving initiatives to upskill and enable customer success in the region on all things informatics across next generation sequencing application. So today she will kindly give her talk on accurate and accelerated analysis solution for Illumina NGS data, and your time is 35 minutes. Mrs. Tan's time is yours.
Thank you. Just bring up the screen. And can I confirm you can see my screen?
Yes, clear. It's clear. Okay.
Thank you very much. It's a pleasure. Thank you for having me on this conference today. I think I'm really excited to take this time to have a great sharing session and really get to learn from the different speakers and the participants in this session. Very excited that this is the inaugural session for the informatics in Indonesia. I've been with Illumina for the past 8 years, and I've been working very closely with the Indonesian users, right, on enabling informatics for NGS. And this is the first time we're really seeing a local conference, and I'm personally extremely excited. So thank you for having me. And yeah, without further ado, let me just get on to the slide proper. So today I'm going to share some updates around the informatics portfolio for Illumina, because I think, you know, as an inaugural session, as well as, you know, all the updates that's really upcoming with the Illumina informatics, we really see that a lot of requests coming in from a lot of our users, right? Asking about what's happening with Illumina informatics. There's so many changes. How can we utilize this platform to really help drive our research and clinical use to a next step? So I think this is where I really want to take this session to achieve that objective. So it's not meant to be technical. So anything that you require technical information, please feel free to reach out and we can always follow up, right? So I think with regards to Illumina informatics, it's always about delivering an answer to the amount of data that's being generated on the sequences through the genomics workflow, right? From sample collection all the way through analysis, sequencing and analysis. And the Illumina analytics really come in way from the sequencing step all the way through downstream analysis. So if you look at a little bit of the direction of Illumina informatics, it's really about building that ecosystem, right? Because we know that the instruments generate a lot of data, right? And it has been scaling so fast over the past 20 years. We've been seeing 100 petabytes of data globally that's been churning, day by day. So we need to ensure that we do our part to invest in solutions that can help our customers go to the next phase of managing data so that they can focus on looking into the interpretation of the data. And that's where the whole directive of Illumina Informatics is driving towards. And I'll go through a little bit of that as a framework over the next few slides. But as you can see, in terms of Illumina Informatics as a common framework, we are building an ecosystem that can really allow all the different components of the genomics workflow to talk to each other. And that really links to the informatics as well. So we want to avoid, right, having a software that only talks to a specific workflow, for example, a cancer-specific workflow or a micro-specific workflow. We want to be able to open up the ecosystem so that they link to each other, that can further incorporate phenotypic information of patients, health records, databasing, and notes taking, etc., and etc. And this is where we are potentially going to build a layer, an architecture, potentially to serve the needs of our users, building on the secondary analysis layer, which are structured around Dragon, as well as different suites that suits different application need. For instance, across the base-based suites that we have that we'll talk about later, the TrueSight software suite pertaining to genetic disease testing, as well as third party. So we are definitely working very closely with the third party solutions that are available to help drive the end-to-end solution for all our customers. And this is definitely in the works, and I'll share a little bit of that later as well. For this session today, I'm going to focus mainly on the second point to the fourth point on this slide here. As you can see, Illumina doesn't only include informatics, bioinformatics from secondary analysis onwards. We recognize and highly value the need for a LIMS platform that really talks to our instrument, that helps capture phenotypic as well as metadata of our samples when the genomics workflow starts. So Clarity LIMS is really that platform to allow the tracking of this information. But for the purpose of this session today, I'm only going to focus mainly on the secondary analysis, which will be from Dragon to base-based suite to TrueSight software suite. Okay. Right, so I'll start off with Dragon, and I'm not sure if everybody have heard about Dragon, but Dragon has really been picking up popularity these days, and particularly because of the works that has been published by Broad Institute. So I guess most of you are very familiar with Broad Institute, having been the gold standard in the genomics informatics world, leading the industry pipelines building for the last 13 years. And in 2019, late 2019, October timeframe, we actually started a collaboration with between Illumina and Broad Institute to enable a new software, or not really a new software, but really a software that we are making available to the general community in 2021. And as you can see on this particular poster that was actually delivered and presented by Broad Institute during the conference in 2020, they actually publicly recognize and confirm the use of the software Dragon in terms of speed, in terms of accuracy for analyzing genome analysis based on human whole genome in their clinical workflow. right? As you can see, these software that we are collaborating and making available and are already available for our users today is the Dragon GATK pipeline. And you can see in their blog, they actually publish accuracy gains that they have done independently with Dragon in blue dots and the GATK best practices in orange. And you can see across the indels and the SNPs precision as well as sensitivity, Dragon outperforming that. And this is something that really was a big shout out by the Broad Institute. And you can see in this follow-up slide that Broad Institute actually showcase in their poster is that Dragon has replaced GATK best practices in their own production workflow. As you can see, it helps with, it's really integrated into their workflow for fast queue generation, map and align, as well as variant calling before it goes into downstream warehousing and tertiary analysis, right? And that is really where we are talking about Dragon today, because we recognize GATK as the industry standard a couple of years back, and Dragon was functionally built around the concept of GATK best practices. But over time, Dragon's, the ethical team that Illumina acquired had really, outperformed in terms of accuracy and speed and performance over GATK best practices that really brought about this partnership that happened in late 2019. And this is something that the Dragon team from our Director of Informatics of Illumina, Severin Catroux, as well as the Broad Institute data scientist, Dr. Eric Banks, who actually came together to really help the community understand that Dragon is really that next era of genomics pipeline that help improve the accuracy as well as the turnaround time for genomic analysis. And he really, Dr. Eric Banks, came forward to really recognize some of the key challenges they had with GATK best practices, which were overcome by the Dragon pipeline, which they have now incorporated into their own production pipeline. So I've put a link here in the slide for people who are interested to really follow up and hear directly from Broad Institute in terms of how much they are valuing Dragon and looking forward to make available Dragon GATK in 2021, right? And I think with these kind of collaboration and really having Dragon, sitting in the production line of the Broad Institute, replacing GATK best practices, what we are seeing today is really Dragon being adopted as the mainstream workflow for the population genomics projects at the national scale. And we are looking at the All of Us program in the US, and this is the latest because we have Genomics England, the UK project that was ongoing for the past couple of years. They have already moved on and converted their pipelines from GATK best practices to Dragon GATK. And mainly because they acknowledge the accuracy gain, right, as well as the improvement over the overall workflow that they are hoping to really drive and scale up the national scale project that they're building, right, across the different nationalities. So this is a press release that was actually shared by the All of Us program, the Genomics Centers, the University of Washington, Broad Institute, and the Baylor College of Medicine actually are using Dragon GATK, which is actually the version of the Dragon 3.4 that many of our customers are already having. In fact, our customers are using a more recent version, 3.7, that has a lot more features and additional performance gains compared to this version. So I think that's really where we want to help update the community around the availability of a pipeline, right, that Broad Institute has recognized and are going towards building a new dynamics for genomics analysis. Right. Dragon is not just something that is very much based on research. It has an extended arm really into the clinical space, really to help drive clinical research into actual diagnostic utility. So Dragon is actually incorporated in 2018 into the Radish Children's Hospital by Dr. Kingsmore, and they were the team that actually obtained 2 times Guinness Book of World Record holdings, right, for the analysis speed. And as you can see from the slide, they were actually able to clock 19.5 hours for an entire workflow from sample collection all the way through reporting. And this is not possible if they had not utilized Dragon, right, which took about 30 minutes at that time. So Dr. Kingsmore has also recognized that Dragon is not only just fast, but has provided improvements in terms of accuracy that really help improve in the overall result. And Dragon really is fast. And I want to give you a little bit of perspective on what I meant by fast. So if you look at Dragon using the version 3 that we have on the left of the graph here, this is based on the germline whole genome workflow at 30x coverage. So if you actually look at how Dragon runs in the blue bars versus the GATK best practices in the purple bar, the overall time we are taking is about less than 25 minutes per genome. And we do see the GATK best practices running at about 10. And in some instances, when I speak to different customers, they were running like 24 hours per sample without optimization or a huge HPC availability. So I think this is really where we want to drive that cost effectiveness of the turnaround time, right, for Dragon. And it's not just for the germline workflow. We also have examples of how the somatic tumor normal workflow has really obtained the turnaround time achievements with Dragon under 2 hours per tumor normal, right, at 100x and 33x respectively. And you can see the comparison against the GATK best practices and Stroker, right, that Dragon consistently has the lowest turnaround time, like the quickest turnaround time compared to the other available workflows. So it is fast, but this is not the only value, because Dragon is really, really prioritized for its accuracy. So what we have done was really a second time Dragon took part in a Precision FDA Truth Challenge, right? It is a challenge that is actually as a standalone competition held by the Precision FDA team. And Dragon actually won 2 categories this year, right? And it was only announced in May. So these 2 categories reports the difficult to map regions using Illumina data and all benchmarking region. So the one thing that's really relevant in this particular innovative approach for Dragon is really having made new features into their already good germline workflow to really help map reads into the difficult to map regions of a genome. And that includes region that crosses 193 clinically relevant genes that were based on truth sets that are derived from either linked or long read sequencing. So these are the real data that we are really very excited about, because having reads that map to these regions of over the 193 clinically relevant genes, it just means that Dragon can now yield much better variant calling accuracy across these genes, which in turn leads to a better diagnostic yield in our clinical trials and clinical research. And this is by far the most accurate and cost-effective way of driving clinical utility, adopting NGS, right? So I think this is really one of the big shout out. And one of the things about the innovations in this Precision FDA Challenge is that some of it has already been incorporated into our latest version that is made available to our customers. So our users are already using these new innovations. And as you can see from the graph on the right, Dragon really are outperforming even the second in class within the participants that enter this challenge by a 28% and 38% fewer errors. Definitely accurate, yet reducing fewer errors compared to the other submissions, right, in these 2 categories, and really help, drive that turnaround time and accuracy over the entire results, right, that drives into reporting. The next thing I wanted to really share is the actual usage of Dragon, right, into diagnostic, right? And in this particular case, Children's Hospital in Los Angeles really incorporated Dragon into their overall workflow and really helped made a positive impact into their clinical management for point of care using their clinical whole exome sequencing. So it took them only about 4 minutes per exome, achieving 99.4 and 99.9 sensitivity and specificity with 100% concordance rate, right, for clinically significant variants. And I think this is really something that I want to call out, right, because this is not just a research tool. It can definitely be something that has been proven and drive towards increasing diagnostic yield. So I do see where the path of Dragon can really help improve healthcare in the long run. So I think this is where showing this information hopefully help, you know, drive some understanding of how Dragon has been performing in the clinical use. So I think one of the things that I really want to shout out is the kind of diversified and versatile pipelines that we actually have within the Dragon suite. So as you can see, it ranges across all this spectrum, and Dragon is really just a 2U server. You know, it doesn't take a whole room of servers. It's really just that small 2U rack, as you can see on the graph on the graphic here. And what is amazing is really the clever way of how the Dragon team actually perform hardware acceleration of the software into this FPGA chip to put it into this PCIe card to lock it into the server. So this is really an integration of a high performance server, maximizing the capacity of the resources you have in a server into your existing infrastructure. And it comes with it all these pipelines that are pre-configured for you, as well as the ability for you to configure the parameters that you need. And if you look across the pipelines, really those in orange are the new pipelines that we have made available, including QC, right? Because we know that a lot of customers are very, very interested and has been asking about enabling QC on Dragon, and we made it available in our recent update. The germline and somatic support, right, for exome whole genome analysis, CNV calling, structure variant calling, repeats, right, STR, short tandem repeats, and expansion hunter, kind of a repeating detection, as well as CYP2D6, which is really a critical pharmacogene that's involved in about 20% of the commonly used drugs across a very broad spectrum of medical discipline. So I think this is really something that we have shown the improvement in the algorithm, really adopting Illumina reads to help with calling difficult to call genes, a critical pharmacogene, highly called out in precision medicine, single cell RNA, as well as trios and cohort analysis for large scale national projects. And really looking at the application, right, this is really a very quick snapshot of what kind of application we cover besides the germline, somatic, across oncology. We also have support for UMI, as well as TMB, MSI, and into the RNA space, right, looking into expression, single cell, as well as metagenomics that I'll share a little bit on because of the current pandemic. On the right, we have a list of all the matrices that comes along with these applications when you run the pipeline automatically. So things like file compression that's going to be made available in the coming quarter, right, in 2021 at the FASTQ level, but FASTQ compression at the BAM alignment level is already available. Your coverage matrix, alignment matrix, human contamination, right? There's already a call out in automatically within the pipeline, ploidy detection, as well as specific QC trimming. Okay, right. Another application that that people are very interested in Dragon is really the cohort analysis that looks into family-based joint calling, as well as large-scale population genetics. And Dragon today can actually perform and support up to 10,000 samples if customers is looking to generate a multi-sample normalized genotype output. And you can see this is generally the framework for getting the GVCF into a new module that Dragon has available today called the GVCF genotyper that really generates that normalized genotype output to help inform what are the variants that are different within that population. And for data that are potentially low quality, we can perform an additional step called the joint genotyper to improve the genotyping for low coverage region. And these in recent versions have seen a 40 times reduction in the time that we actually enable our customers to do. So you can see that in now, if you have 1,000 whole genome, you can actually generate your joint call in 24 hours on a single Dragon, which will potentially take months if you were to perform using GATK best practices. So this is a mass achievement that we are really hoping to build and help as a key ask from all our national scale project KOLs. And last but not least, as a key feature for Dragon is really enabling the data compression, right, feature that we are really helping a lot of our customers save, not just the turnaround time, as well as operational costs, storage costs. And that is important to us because we have already allowed the use of alignment file compression, but now we are taking the additional step, bringing up the tier to now compress FASTQ files to save on the file size. As you can see across 3 different RNA samples, we see about 5 times reduction, really saving the cost of your data storage while preserving data integrity and increasing increasing the file transfer by reducing the bandwidth required, right, in through file transfer medium. Okay. Well, that is Dragon. And the next thing I really want to give an update on is really on the base-based sequence hub. And for customers who have been with Illumina for some time, I'm sure you have heard of this base-based sequence hub platform that's really aimed at helping a lot of new to NGS customers get up to speed very, very quickly from day one at a snap of the fingers, right? Pushing their data through to reporting so that they get their variants, their insights into that data as quick as possible. So base-based doesn't require any installation, right? It is optimally used for sharing and collaboration across global users, as well as in-country between different collaborators. It has a suite of analysis suite that I will talk about a little bit today with a focus into the informatics beat, as well as the integration for advanced informatics people who are already, leveraging the kind of genomics informatics today.
Mrs. Tan, you have another 10 minutes left. Thank you.
Thank you. Yep. So really, I want to put this slide here to show that base-based as a data storage is really available now as a new feature to really help customers to do long-term storage while really preserving the operational costs, as well as the types of data analysis options there. So you can see it's really point and click, very easy for customers to achieve different kinds of objectives across QC, detection, as well as identification. Right. So what I wanted to show here is really turnaround time. And you can see for the Dragon feature that we're included in the base-based platform, we are seeing at least 10 times improvement, right, in terms of turnaround time, which in turn save you costs for the operations, as well as our very recent availability of our emergency use kit for COVID testing. We have availability of the Dragon, really help achieve that high sensitivity and specificity with the turnaround time of 1 to 3 hours, depending on the number of samples you are running through the workflow. But this is really generating that report for SARS-CoV-2 detection. And an example of a report I've made available here, really to help you see which are the samples that are detected versus not. We also have made available new pipelines called the Dragon RNA pathogen detection pipeline. I'll not go through in details here, but really as a key matrix to really help share the speed, right? It's 190 times faster than a top hat alignment that really help, the speed up the detection of SARS-CoV-2 in the samples application that you've chosen, as well as the reports that are being generated to confirm or infer the identity of your samples, as well as the support for shotgun metagenomics, really to help provide Dragon as the aligner to perform human host removal, as well as report generation through visual graphics, as well as actual matrix and the consensus FASTA, right, for the genome generation. We also partner with third-party solutions, and this is an example where we have a rat detection solution for respiratory pathogen and antimicrobial resistant genes that we partner with ID by DNA, available on BaseSpace to really help generate those reports, as well as mutations that really inform for micro testing of the patients in a clinical setting. And this table here really shows you different aspects of the report that BaseSpace ID by DNA Explify report detailed into, with the different scorings and the explanation and the interpretation of the results. We also have updates into the oncology space, really driving Dragon into the secondary analysis and partnering with PeriodDX CGW software to really drive that report generation. As you can see at the point of click, really having the 500 over genes span across 1,000 variants, typically interrogated down to a couple of meaningful variants and a final report. And this is an example of a report and highly customizable depending on the user need to really serve the need of oncology testing requirement. Next is really the tertiary space, and these I wouldn't dive too deep in there, but really want to share the offering of a 6 months free access to a correlation engine on BaseSpace that help provide high quality curated public data knowledge base to really derive patterns in your data through aggregation and doing meta-analysis over 23,000 scientific studies that are shared by different collaborators, right? And I think one of the key things that we really want to enhance in the COVID host response era, particularly in this pandemic, is really leveraging information that we have across thousands of studies that are shared by the scientists to compare the coronavirus infection across the different animals and humans, and really be able to allow you to ask those questions around what are the host response, how they relate to outcomes, and what role does the correlation engine play is really to routinely update this information and the database so that you have a pre-packaged data for the SARS-CoV-2 research to quickly contribute and drive insights into your queries in real time. An example of the kind of interface that we are seeing here is really a lung biopsy that's taken from a COVID sample versus a healthy sample. And you can see specific genes that are being called out, that the ACE gene as a viral receptor for SARS-CoV-2 that people are well aware of has been called out, that has been found within this sample with a specific fold change, control expression, and quality score. And you look into the specific body atlas for the most correlated tissues, etc., and etc. It's really about informing the aggregation of the data, looking into the host response of the data group that you are researching on. And the next tertiary interpretation tool is really on the variant interpreter side that most of you are probably aware of, but I just want to enhance on the knowledge that we are continuing to really build this software for cancer research customers, for genetic disease customers, for research, right? So I think I will quickly move on in view of time to the last bit of my presentation, which is particularly focused into genetic disease testing, right? It's based on the TrueSight software suite, the latest software that we have available that really take in your sample, right, that you receive from the sequencer and run it through end to end without any further interrogation midway. And this is something that is like a turnkey solution, really to help customers look into whole genome, whole exome variants that are scaling into the thousands and the millions, and really help you to triage and look and visualize and interpret the variants across this spectrum of mutational classes. And I put in this slide here some of the key software features that really help drive the utility managing whole genome and whole exome variants in a clinical setting, right? Running it through an entire workflow, incorporating the Dragon solution to look into variant prioritization, annotation, building your own database, right, on variants that uniquely only your lab has, as well as the ability for you to learn from databases that you can put in or collaborate with other entities. This is just the interface of how it looks like, really to help inform our users that you do not need any programming knowledge to use this software. It's visual drag and drop to really help build your own pipeline on how you want your variant filtering to be progressed. And then you look into building your own database through visuals, spanning across the coverage, the databases that you choose or provided by the platform, as well as reporting the final report per patient. And last but not least, I just want to quickly share that we do have the National Health Research Institute in Taiwan that has really seen the true adoption of the TrueSight software suite, 24% increase in diagnostic yield based on 52 trios epilepsy whole genome sequencing samples that they would not have been achieved if they have not adopted TrueSight software suite in such a short, quick turnaround time. Okay. And my last slide here is really a quick summary of the kind of components that I've talked about, really going more into the Dragon GATK collaboration update, the suite and pipeline available to help you drive genomic analysis, the kind of data management platform, as well as the additional pipelines that we made available on the Base Sequence Hub to support the pandemic, as well as into the oncology space. Tertiary analysis tool that we have available to really help drive further tertiary research, as well as interpretation and reporting for research into genetics and oncology. And finally, TrueSight software suite to look into whole genome and whole exome genetic disease testing as a turnkey solution to drive the utility and increase diagnostic yield. I hope I'm on time, and that's all I have. Thank you very much.
You are on time, actually. Okay. Thank you, Mrs. Tan, for your very highly valuable information on the technique of Dragon, especially. And then, you know, during the pandemic, the speed of the data available can resolve pandemic very fast, I think. And I was thinking that Illumina also give a big contribution on this pandemic issue. Yeah. And there are several questions from the audience. I would like to see first. The first question is the proprietary part of Dragon necessary for the accurate results, or is that for platform-specific needs only, and the open source used in hybrid with the GATK will result in the fast time and higher accuracy from GATK? From Sabrina, Queen Mary University of London.
Thank you, Sabrina. Thanks for the question. So the accuracy gain in the Dragon pipeline is really something that has been built on the concept of GATK best practices. And this is currently a proprietary software that we, as a collaborative effort from Broad Institute, will be making open source, as you mentioned, in 2021, right? So the reason in terms of how the Dragon pipeline has outperformed the accuracy of JATK best practices really comes in several factors, and we have a data sheet that we can always follow up to share details with you in terms of the mapping, foreign read detection, the way base quality scores are estimated on JATK best practices is sort of outdated versus the actual software generation of base quality scores across sequences today. And of course, these are some of those factors that really help improve the achievements on the accuracy side that are also acknowledged by the Broad Institute. And I think one of the things that the Broad Institute really, see the value is really into the kind of graph construction on the haplotype caller that is unique to the Dragon pipeline that will be adopted by the Broad JATK, Dragon JATK software that will be made available in the next quarter. And these algorithms will soon be more visible into the community on how these actually works. So I'm also looking forward to having that available, so that our aim is really to help the general community access a more accurate software so that people are generating more accurate data sets for data sharing in the long term.
Okay. Thank you. Another question, okay, from Sabrina as well. I think this is the continuation from the previous question. How well can Dragon work with the fragmented alignment when we have low coverage sequencing or doing different non-model species? How will it performs?
Yeah, so Dragon is developed mainly as a start to really help drive whole genome sequencing utility. And as you start to see our expansion into whole exome sequencing, as well as into the somatic space and the different other applications. So in terms of fragmented data set, I'm not quite sure whether you are referring mainly to whole genome applications or targeted applications, because it also depends on, maybe you're referring to oncology, FFP samples, right? So I think to a certain extent, the quality of the sample, right, how fragmented the data will impact the overall data quality of, it's not so much of Dragon handling it. It's really because if your data is highly fragmented, the library size are much smaller, you end up only sequencing much smaller fragments of your data. And when that passes into Dragon, Dragon continues to do its job in terms of mapping these shorter reads across the genome and find the best match and generates the data. But of course, it also then brings into other features such as the coverage information, right? And across the breadth of the genome. In Dragon Suite today, it still do not have support for targeted panels like implicit specific assays, which we are making available in 2021 as well, to really help address that smaller target regions within the entire human genome, and then validate that against somatic samples as well. So I would say that is something that will be is already on the roadmap and will be made available next year. I hope that answers your question.
Yeah. Thank you. So I think this is the end of time. Once more, I would like to thank to Mrs. Tan for your enlightenment talk on Illumina and its technology. And then I hope that it still continue to help and to boost the information of COVID-19 to solve the problem of the pandemic for sure. And then on behalf of the organizer, we will end for the keynote first session, and then I would like to thank to Professor Ahmad, Professor Fahrur, and Mrs. Tan for very nice talk and very valuable discussion. And for all of you, if you would like to discuss more detail, you can contact directly, I guess, to every speakers via email, or probably start with the little chat as I did with Professor Fahrul. And thank you very much, and I'll give it back to this organizer. Please.
Thank you very much.
The keynote talk, the fourth keynote talk, we will continue after the break. And then get the message from the IT. We will have a photo session again. This is the third photo session. And then I will the IT. Please show your photo and give the best.
Thank you. Let's get ready for the next photo session. I will help with the management of the photo session. Please get your camera ready. I'll take now. 1, 2, 3. All right, another one. 1, 2, 3. Right. Very nice. The next one. 1, 2, 3. The fourth room. 1, 2, 3. Okay, the last one. 1, 2, 3. And another one. 1, 2, 3. All right. Thank you, everyone. Thank you.
Okay.
Thank you. I'll return to MC.
Okay, thank you very much. And then we will have a break up to 12:30 for the last keynote talk from Professor Kholis Abdurrachim from SGU. And then I think we can start for the break. Thank you very much for everyone. Then we will come back at 12:30.
Honorable moderator, distinguished speakers and guests, and all the participants. Thank you very much for having me. It is an honor for me to be here to give a keynote address on some particular issues, especially on bioinformatics development and also biodiversity-based research. So since this is a keynote address, I will not specifically talk about very technical things, but it will be more into strategic perspective on the development of bioinformatics and also, of course, the direction of our research in the future, especially related to our natural resources, which is biodiversity. So let me please share my talk today. The title of my talk will be okay. Is my voice audible? And also you can see the presentation, Prof. Khomeini? Okay, so the topics is, the title of the talk today is the roles of bioinformatics in accelerating biodiversity-based research in Indonesia. So I will touch a bit about, please allow me briefly to introduce my university. It's a Swiss German university, although the name is European name, but it's actually more established in Indonesia in 2000, which is the first international university in Indonesia as a collaboration between Indonesian, Germany, and Switzerland government. So it is a non-profit foundation in Indonesian law. We have 3 faculties, 11 bachelor degrees and 3 master degrees, where I am now in the Department of Biomedical Engineering, which is actually the first biomedical engineering program in Indonesia. The lecture is conducted in English, but students have to take 4 semesters of German because they have to go abroad to Germany or Switzerland to do internship and also 1 semester course there. We have now currently around 180 full-time and 90 part-time faculty members. Ladies and gentlemen, this is a brief outlines of my talk today. I will talk a bit about an overview of Indonesian biodiversity, some potential and threats, of course, and also research priority in Indonesia based on the Ministry of Research and Technology, and the high throughput data generating experiments and data analysis, which we'll be talking more about opportunities and challenges. And of course, I will elaborate more on my project, which is the development of Indonesian natural products or extract library in PAL for drug discovery. And last is the conclusion and recommendation. Before I start, I would like to elaborate more in this slide because somehow the journey shape where and what I've been doing right now. As Prof. Khomeini mentioned about about my background. So after I graduated from IPB University, I moved to Malaysia, in which I learned about molecular biology. And for the first time, which is in the late 90s, I was exposed to bioinformatics, which is around 1999 or 2000. And also, my lab is actually the first lab or the first study program where the first study program of bioinformatics was established in Malaysia, which is in University of Malaya. That is when I got in touch with bioinformatics. But again, I cannot claim that I'm an expert in bioinformatics because I just use bioinformatics as a tool. As maybe I can interpret some of bioinformatics data, but I've been using this for entire of my research, both my either in master degree, PhD, and also postdoctoral degree. That's why, as I mentioned, this somehow shaped my journey throughout my career right now. So from Malaysia, I moved to United States, where I did my PhD in biochemistry and also my postdoctoral at Yale University. And again, I learned a lot about this journey, and I tried to compare with Indonesia. And eventually I end up, before I return to Indonesia, I end up in Saudi Arabia. So if you compare between the three countries or the four countries, Indonesia, Malaysia, United States, and Saudi Arabia, you will see a lot different. For example, in United States, I know everyone here know about NCBI. Actually, United States, in terms of biodiversity, they don't have much about biodiversity. So they're not even in the list of the top 10 mega biodiverse country in the world. But you know that the NCBI absorb almost everything in the world, any research, you name it. Then they utilize those information to build so many things, yeah, include drugs and vaccine and so on and so forth. While you compare with Saudi Arabia, literally there is nothing there. Everywhere you go, you will see the desert. Yeah. So when I return to Indonesia, I see how wonderful our country is. We are so blessed with so much opportunities and in richness of so-called biodiversity. So that is why when I started join University in Indonesia, first I initiate what's so-called, I still remember in 2013, we have our first NGS seminar. Yeah, at that time also PT Pandu was supporting us. So PT Pandu always support us from very early on. So thank you very much to PT Pandu. I'm not promoting, but indeed they support us very much throughout our journey. So that is where MABI or ISBB is actually seeded. Yeah, the seed of bioinformatics Indonesia is planted. Yeah. So in 2014, we had our first workshop in bioinformatics. When I said bioinformatics, it will include, of course, computational biology. Yeah, where some of the lecturers here, Prof. Han, Prof. Setia, Prof. Bimo, help us in delivering the subjects. Yeah, it was 2014 when I was still actually I was still not at SGU yet. But again, in 2014, I had the opportunity to establish the first and maybe until now the only one bioinformatics study program in Indonesia, which is at I3L in 2014. But later then in 2015, I then joined SGU. So all these journeys really give me a very strong reason why I try very hard, actually, of course with other colleagues, to really promote bioinformatics. Yeah, although as I mentioned, bioinformatics is not one of my strengths because I'm a biochemist, I'm a molecular biologist, but I use a lot and I was exposed to bioinformatics since about 2 decades ago. So that's about the actually today. Yeah, this is actually quite emotional. Prof. Khomeini, I need to share with you all this year because this will be my last day of being a president of MABI. That's something, an organization that I tried to nurture since 4 years ago. So let's talk a bit about Indonesian biodiversity. I'm sure you all know about our strength, our biodiversity, but we just kind of refresh our memory how rich we are in terms of biodiversity. But of course, all potential always comes with threats. So let's see our potential. Indonesia has, although it is an international conference, but I know that my talk will be even more into how to build Indonesia as a great nation, because we all these tremendous resources. For example, population. We have nearly now about 280 million people. So everything we have, everything people sell, any companies, yeah, Indonesia, they will become rich people. You name it. All pharmaceutical companies, big pharma companies, Pfizer, Novartis, Sanofi, everything is here. Yeah, everybody is here. Because of what? They don't do research, they just sell products. This is something that we have to really think about in the next generation. Are we going to be just a consumer, or we are going to be a producer at one point? And of course, geographical location that really shape our biodiversity. We are one of the richest biodiversity in the country, in the world, yeah, in terms of both on land and also in ocean. And again, about natural resources. So this is just a glimpse about how rich we are, yeah, in many ways, yeah. Even we have about more than 300 ethnic groups, yeah. And of course, Indonesia here is listed as 1/4 of the mega biodiverse country in the world, because we have so much. In fact, especially for fish, we have about 25% fish population. Yeah, so we have so much, so many plants, so many endemic species, and about 6,000 species used for jamu. This is why what I'm trying with my colleagues, of course, trying to promote more is actually the biomedicinal plants in Indonesia, because it's not that easy to find resources if we want to search or find for any literature or any database that is mentioning about uh, everything that is derived from Indonesian biodiversity. That is why I try to more focus on the natural product library, which I will talk a bit more later. So as we all know that, for example, plants, we know that plants is used for so many purposes. Yeah, here I have some data from botanical garden in the UK. We have for medicinal plants, there are close to 20,000 plants with issues for medicine. We also know that plants used for materials such as papers, and many other materials. And of course, environmental uses, human food, gene sources, animal food, poisons, fuels, social uses, and any other uses. So these are just some of very few mention of documented uses of plants. Not to mention about biodiversity in the oceans, not to mention about biodiversity in terms of microbes, which are now very emerging. Yeah, marine microbes is one of the top leading area of research, not only in the world, but also in Indonesia. The reason I mentioned all this is because later on, we have to realize, we have to understand that bioinformatics is just a tool. Yeah, and at the end of the day, the research will depend on the ideas, what ideas that we have that is required by informatics. So please keep that in mind. Yeah. So research ideas is actually more important than just tools, which is the bioinformatics in this case. Yeah. So if we, for example, currently we are facing a tremendous burden on COVID-19 impact. Yeah, even before, long time before COVID, we've been struggling with dengue virus, for example, malaria. So now we have COVID. But if you think about the potential, the economical potential behind that, even only for dengue virus, for example, we can really estimate about $4 billion only for diagnostic. Well, we all know that vaccine for dengue virus is not established yet, and of course, for other also is not yet, including now for COVID is still under ongoing. Yeah. So having all this, what I'm trying to say is that we have so much potential in doing research, of course, and also in deriving or generating money or incomes for our country. So you can imagine if we are able to generate this amount of economic, yeah, for only from one single diagnostic kit for dengue, for example, you can imagine how much money we can generate from different types of diagnostic kits, for example. Yeah, unfortunately, until now, yeah, not to I'm aware of, to the best of now my knowledge, dengue kit, diagnostic kit dengue is actually produced by foreigners, yeah, from overseas. We import from overseas. Although now we know that serotypes of dengue from Indonesia is quite different compared to serotypes of dengue found elsewhere. So this is actually open the opportunity for us to develop new diagnostic for dengue. Not to mention about vaccine, drugs, and also, of course, other diseases such as COVID-19, tuberculosis, and others. So this, again, I would like to remind all my fellow citizens that the future is actually in here in Indonesia, if as long as we can utilize this properly. So in addition to potential, yeah, our biodiversity also possess a very big threats. Yeah, we know that almost all kind of microbial pathogens, either viruses, bacteria, fungus, parasites, and many other microbial pathogens exist in Indonesia. And up to now, we were not able, yeah, not a single one, again, to I'm aware of, generate something that can be utilized to overcome these issues. Yeah, for example, malaria. Yeah. And now, not only the old drugs or the old vaccine, in fact, for tuberculosis doesn't work properly anymore. Now already coming, there are so many other new strains, new species that are now resistant to different types of antibiotics, for example. So this is a very major issues nowadays that we are facing. So in addition to what's the re-emerging disease, we also have the re-emerging pathogens that we have to deal with. So this is all up to us later on in the future. WHO in 2017 released about 12 bacteria for which new antibiotics are urgently needed. Yeah, they categorize into different types: critical, high, and medium. And one of them is actually the one that I work with is Pseudomonas aeruginosa. Is now almost resistant to all kind of antibiotics. Yeah, so this is a tough challenge that we are facing. And apparently, WHO also have a report, although it's quite old, in 1996, that every 20 years there are 30 new diseases emerge. So we are really dealing with competing with the pathogens, yeah, in a very fast phase, yeah. So before I go deeper into what we are going to talk about more today, which is about Natural Product Library, let me just share briefly about the research priority in Indonesia currently. Yeah. So this is again, sorry, I skipped one slide. This is again another issues we are dealing with, which is the national security issues. We are not ready yet for biosafety, biodefense, and biotourism, as we are now realizing we are short of any facilities in terms of facing with handling or dealing with COVID-19, for example. Thanks God there are new labs now established, although I was informed that many labs is not following the regulation yet in terms of biosafety level, for example. Not to mention about biodefense and bioterrorisms. So we are really need put some very big effort to deal with this issue. So let's briefly share about the research priority in Indonesia. Indonesian government released what's so-called Indonesian Grand Research Plan for 2015 until 2044. As you can see here is that the natural resources-based applied research is actually in the top priority. Although now the second term, which is 2020-24, but I believe that it's still the natural resources based applied research is still dominating in the next few years. Yeah. So from time to time, is of course the priority change, but if you see, the natural resources based applied research is still the top priority of the government funding. So again, this is I share with you this just to again remind all of us that we are a very big country actually in many ways. Yeah. In fact, in terms of universities, we have around 4,000 universities. If you can imagine, every single university work on, or any researcher in one university works on natural products based research. So you can imagine how much resources that we have, yeah, or we have to do this research, and how much information that we can really gather and give a very more minimal information in terms of biological information, yeah, in different ways. So now let's talk about high throughput data generating experiments and data analysis. Yeah, here I would like to address some points that in order to be able to have or to utilize bioinformatics, yeah, we are in need of what's so-called bioinformatics is a tool, okay, to use for generating or analyzing high throughput data. So in order to do that, yeah, we have to have experiments that are producing high throughput data generating experiments. But what I'm trying to say here is that why we have to focus on our biodiversity, yeah, our biodiversity as source of bioinformatics. You can imagine bioinformatics is not very old things introduced in Indonesia, right? So it means that other people, other countries already established in terms of tools and many other research in bioinformatics. So if we are trying to lead in the area, so there is no any other way, but we have to use our own biodiversity resources. I'm not saying that we should not collaborate with other people. No, I'm not saying that. But we have to be aware of our resources, yeah, before somebody else utilize it. Because if we use this, for example, dengue virus, if we, let's say, establish a diagnostic kits for dengue virus, of course, the accuracy of our dengue kits will be much better if we use our own resources in terms our own by dengue virus originated from Indonesia or serotype, Indonesian serotypes, compared to serotype that is used or established or developed by other countries. So this is again how is a way how we will lead in terms of biodiversity research. So please don't try to compete with other people, especially those who are in the developed countries when it comes to research. But we use our strength, which is our own biodiversity. Yeah, for example, now, I always talk to our colleagues in different groups, yeah, MABI or IPSB or any other scientific organization. For example, COVID-19. Actually, it will be very good. It will be best if we can generate our own vaccine come from COVID-19 found in Indonesia. Because I'm aware of, yeah, out of 20 sequence that sent to a G site, 9 of them already mutated. Yeah, mutated already. Yeah, so we already know that. Yeah, so I'm afraid that if we, let's say, use vaccine generated from SARS-CoV-2, which is different from what we have here, it will not be very useful. Yeah, so that is from, of course, from scientific point of view. Yeah. So let's just spend on this slide quite a bit. Why I mentioned about biodiversity as source of bioinformatics. We all know that we have so many organisms, you name it, mammals, plants, birds, fish, fungus, microbes, in fact, even human. Yeah, we have so much potential to research on. And then from that research, we can generate so much biological data. Yeah, biological experiment sequences, DNA, RNAs, proteins, lipids, metabolites, and so on and so forth. There are so many biological data generated from our own research. Yeah, forget about other organism that doesn't exist in Indonesia. Yeah, we have more than enough if you want to really utilize our own resources. Then come omics. Yeah, from this biological data, we generated different types of omics. Yeah, genomics, proteomics, lipidomics, and so on and so forth. Yeah, not to mention again about single cells. We have not talked yet about single cells in Indonesia. Yeah, I believe to the best of my knowledge. Yeah, maybe inform me if someone work on this, but to the best of knowledge, there is no one yet work on single cells. Yeah, we still only work on organisms. In fact, until now, yeah, we have not really generate a complete genomic sequence yet from a single organisms found in Indonesia. And actually, from one single data, omics by genomic data, yeah, if we do some sequencing, for example, if we do some sequencing, for example, from genomics, there are as much as petabytes data, a very big data generated from only genomics alone. Yeah, not to mention about proteomics, lipidomics, and so on and so forth. So we have so much data to work with. Yeah, and that is where the bioinformatics come to play. Yeah, we must use bioinformatics to analyze so much data. Yeah, it is impossible to use only simple technique or method to analyze so much data. And then by using bioinformatics, then we can generate so much biological information. Yeah, I always feel the need to explain to in many occasions to Indonesian audience how important bioinformatics is, because until now, yeah, I have not seen that bioinformatics become a center or become a focus or the priority of university, for example, and not to mention about government. But at least anyone here can implement this in a very small scope, for example, in the department, faculty, or university, in fact, yeah, to really start focusing on bioinformatics. Yeah, because until now, I'm sure there is only one study program in informatics in Indonesia. Yeah, I don't know about other university. I learned that IPB have bioinformatics not study program yet. Yeah, I think. Yeah, but again, this takes a lot of effort. Yeah, collaboration effort for all of us to work with together. So nowadays is the big data era. Big data means big revenue. So it's generate so much data. Yeah, even here, Sanger 2009 mentioned about 7 petabytes of stored information for genomics alone. Yeah, so it means here one sequencing data set needs about 2 terabyte sequencing only. Yeah, we have not talked about annotation and so on and so forth. So again, not to mention about single cell omics. Yeah, in 2025 there will be a revenue reached by 5.32 billion. Yeah, so I learned a bit about single cell. So we still now talk about whole organisms, but if we want to be more into detail, then we have to see to the single cell omics. So so you can imagine how many cell in human being. Yeah, so we need to learn. So you can calculate by yourself. Yeah, how much data, how much money can be generated from single cell. Yeah. Okay, this is for youngsters, students, yeah, who are in searching for career in the next year or so. That don't worry, career bioinformatics is widely open. Even there are so many career in bioinformatics. Yeah, 10,000 position vacancies in bioinformatics in Southeast Asia alone, in Asia Pacific alone. Yeah, so don't worry. So, having all said this, I would say that bioinformatics as a necessity for Indonesia. Yeah, if and only if we want to become the future. Yeah, the future belongs to bioinformatics. And if we really focus on or give the priority to bioinformatics, I'm very sure that the future will belongs to Indonesia. Yeah, this is all up to us now. So, of course, in every effort, there is always challenges. Yeah, and there are many major challenges when we talk about bioinformatics in Indonesia. First and foremost, of course, still up to now, there is no grand strategy for biodiversity-based research in Indonesia. Yeah, there is no policy. Yeah, on how, on what we have to work on. Yeah, of course, government give the liberty to us to do anything we want. Yeah, but that's not always good because we have a very limited resources. So we have to work smart, collaboratively, and with a good coordination. So because it will lead to different issues, we have we need research direction issues. We need coordination, communication, and collaboration. We also need facilities, sharing facilities. Yeah, we also lack of sustainable and adequate research funding. Yeah, it leads to disconnected project. I personally experienced this disconnected project. Yeah, especially during this COVID-19. Yeah, and also lack of lack of institutional. Yeah, we don't have enough equipment and supplies. We have also limited human resources, and we don't have national centers that will eventually become the umbrella of our activities. So, in short, I would put that challenges in implementing bioinformatics is that bioinformatics is just a tool. Unfortunately, yeah, it needs high throughput data, which is generated from high throughput data generating experiments. It will take a lot of investment, of course, and it also need more infrastructure and facilities. Yeah, this again, we have to deliver this message to our government to put more priority on bioinformatics. So I tried to search a bit about research activities in bioinformatics in Indonesia, and I end up with all this data. Yeah.
5 minutes.
Okay, 5 minutes. Thank you, Prof.
Yeah.
So this is just some very little. Yeah. So you can imagine from a very big country like Indonesia, we have a very limited research ongoing in terms of genomics, proteomics, metabolomics, transcriptomics, and so on and so forth. Not to mention medical informatics and system biology. Yeah. And out of this very little research, and we also sometimes do things overlapping. Yeah. So this takes some our resources as well. Yeah. So give me in the last 5 minutes talk about this Indonesian that I'm been working for in the last few years, development of Indonesian natural products or extract library. The impel is already registered, not only registered but also granted the copyright. Yeah, in the last year, this is actually we try to build the database for Indonesian natural product library. So the idea is come from the limited drug discovery, and we have to come up with solution that we can somehow accelerate the drug discovery for Indonesian biodiversity. Yeah, which is basically by collecting, all resources we have, and then we store it in our database. Yeah, this is a glimpse about natural products facts and development. The word FDA actually approved more than 50% drugs and also more than 75% of anti-cancer drugs, which are generated from natural products or their derivatives. So please put aside the controversial between conventional drugs and traditional drugs, because actually almost all of them generated from natural products. Okay. So this is again some information about companies that they work only collecting fractions. New York, Albany have 300,000 fractions. NCI, very interesting. That is the reason why we have this 75% of anti-cancer drugs actually derived from NCI collection of 230,000 unique extracts. They collect from all over the world. Yeah, they have the database. They take the database, and also they get the physical extracts there, which actually we don't need the rest of the world in Indonesia. We have everything here. So here again, if I mention point here, yeah, NCI, this is published in 2018, I believe. Yeah, 230,000 unique extract from all over the world. Yeah. So By definition, nature product is a collection of nature products containing active compounds used for screening process of target biology. Don't be surprised. I believe all big pharma companies such as GSK, I took this from GSK, they have this compounds library. Even there are companies they need director for compound management, because you know how much the compounds they have, the variety of compounds. So they just do collection of compounds and they do screening. Yeah. So the Indonesian Nature Products Library, INPAL, is a repository linked databases of Indonesian biodiversity with known function. Okay, please keep that in mind, with known function, especially for drug discovery purpose. So the two main objectives, which the first is database, of course, and the second is the repository, which is the unique identity of INPAL. So we don't have only database, but our database is linked with the physical repository. Okay, so this is the role of INPAL, a window to Indonesian-based biodiversity research, a platform that links the virtual and physical repository of Indonesian biodiversity. The INPAL will be much more useful if, let's say, all Indonesian researchers, deposit all their data here. Yeah, anything related to Indonesian-based biodiversity, you can put here at INPAL. So don't put anymore in NCBI. Well, I know INPAL cannot compete with NCBI. Every one of us will be much more happier if we can put our database in NCBI and all those EBI, for example. Yeah, but again, it takes our collaboration to promote all this what we have here. Okay, so this is some important features of INPAL: database specific species identification. In short, there will be so much data that can be generated from INPAL. Yeah, this is just the data. Yeah, we will have specific species identification, geographical location, method development, extract yields, and so on and so forth. And of course, all those biological assays. Yeah, all this data we can store in INPAL. For what? For later on, someone wants to know about some things, want to learn more, study more, they can refer to this INPAL, and then we can utilize it for our own good. Yeah, just briefly about my research. I use extract mangrove model, an ideal model for extract library, because we know that Indonesia is one of the richest countries in mangrove. In fact, we have 23% of total mangrove wood forest in the world. Yeah, so we have so much mangroves. So this is again the mangrove habitats in Indonesia, very much. There are so many things that we can generate from sampling, packaging, species identification, for example. So yeah, we will have this. Yeah, this is we have also the protocols in how to do the research. So we can store in INPAL. And this is just some example of my research. This is to show again, I like this slide very much because it shows us how rich our country is in terms of chemical compounds and so on. Yeah, only from mangrove. Yeah. So this is the phytochemical analysis, and then this is the appearance of INPAL. Yeah, INPAL like this. Yeah, and then this is the accessing library where we have the NCI. This is the USDA. Yeah, the US, they have the ability to list every plants in their own state. Yeah, what we don't have. By INPAL we will have this. This is from Campbell, MCBI. This is from Knapsack, Japan. Yeah, and again, it will have a very big impact if we do this research. Yeah, in terms of environmental, social, and economical impact. So this is what I did is search for antibacterial and anti-cancer sources, and this work has been published, yeah, in different journals. And as a conclusion, we are in short, we are very rich in everything, and we can so much do out of this. And please help INPAL also in promoting this biodiversity database, because it will need your collaboration. And again, the action plan recommendation, the high throughput screening technology, we have to have this in order to find drugs, to search drugs, and we have to collaborate. That's the key point. And this is the last but not least is the Indonesian that we recommend. Yeah, somehow, someday we have to have what's so-called Indonesian National Center for Biomatics. You can put the name anything you want, but the thing is that we have to have this in our future. And actually, this is one of the main goal of MABI. So this is some of my project I'm shining with Malaysia, which actually out of my expectation, they are very excited to work with us. This is some of my work in Malaysia, developing TB and also dengue from mangrove species. And please allow me, Prof, just 2 minutes. Do I have 2 minutes? Or finish already the time? Yeah. Okay, then we can just skip this.
Universiti Malaysia Sabah UMS dengan kerjasama Swiss German University SGU di Indonesia sedang menjalankan penyelidikan mengenai kaedah mengurangkan jangkitan penyakit tuberkulosis TB dalam usaha menangani peningkatan kritikal selain penularan penyakit TB di negeri ini. Kerjasama penyelidikan bernilai RM400,000 itu masing-masing mendapat pembiayaan grant sepadan UMS berjumlah RM200,000 dan RM200,000 daripada SGU bagi tempoh 2 tahun bermula tahun ini.
Menurut Pengarah Institut Penyelidikan Bioteknologi UMS, Dr. Zarina Amin, kerjasama ini hasil daripada pertemuan dalam simposium
Hello? 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 Hello?
Ya Pak, bisa didengar.
Bisa
Yeah.
dengar? Okay,
Yeah.
thank you. Yeah. So,
Thank you, Kholis.
yes.
I'm so sorry. There's some technical misunderstanding actually.
It's
So,
okay. It's okay. Yeah. So,
okay, thank you very much for your very interesting topic, especially on the diversity of Indonesia actually. And then as Indonesian, we have to realize that how diverse we are, from human to microbe. And then one of the weaknesses, I think we have to admit it, that we still very not pay attention on this mega biodiversity in Indonesia. So I think from this occasion, I think it's very good to start to pay attention on the how we explore and how to utilize this potential, this potency of our diversity. So we have two questions due to the time, limited time. So I would like to from Muhammad Naufal, yeah, no, from Sabrina Gita, yeah. She asked about what are the work needed for our human resources to be able to harvest info from all those biodiversity and data, and how is the state of human resources education around this field? From Sabrina, Queen Mary University of London.
thank you very much for the question. Let me just answer straight. Yeah. So this is a very good question. So what are the work needed for our human resources to be able to harvest into from all those biodiversity and data? So from what I mentioned in my talk, there are of course some important issues that we are facing. First, of course, the facilities, the equipment, the experiments. Well, first, the grand strategy. There should be a grand strategy, the direction from the government on what issues that we have to focus on first, the priority. Second, there should be a national center. Yeah, we have to have national center that will coordinate. Yeah, will coordinate, communicate. So our research source is not going to wasteful resources, because sometimes we do overlap work. Yeah, we don't even know some other colleague works, and then we do the same work, and so on and so forth. Of course, we have first the body, the instruments, the government, and also later on the facilities, and also later on the human resources. Yeah, try to be more focused. Yeah, try to be consistent in doing things. That is what the developed countries do. Yeah, from the very on of their career, they do of very similar things. So we don't just jump from one work to another work. And of course, we have to collaborate. Yeah, we cannot do things by ourselves. We must collaborate. Yeah, if there is no collaboration, there will be no success. Yeah, we cannot just live in a very small nutshell without knowing other people's, because for example, especially in bioinformatics, a single person can do bioinformatics from A to Z, because bioinformatics again is just a tool. You cannot stop the work by saying this is the putative active site without doing the real experiment. Yeah. Because that's just the first clue. The first clue, you cannot just stop at the first clue. You need to continue. And somehow, some way, yeah, we don't have that. Sometimes we don't have that ability. Or vice versa. The people who work with, for example, bench work, they don't know about bioinformatics. So we have to work together. So by collaborating all this, to be honest with you, all my work is through collaboration. Yeah, I don't have, SGU is a very small university. We don't have all the facilities to work with. But by having collaboration, we will able to do a lot of things. Okay, so try to focus on things and work on this consistently. Okay, I think that for.
Okay, thank you very much, Dr. Kholis. The next question is about the source of the NCBI. Yeah, according to you, can Indonesia make a source data like NCBI? If yes, what should we do, and who is responsible for this thing?
Yes.
And I think this is a very nice question.
Yes, it is. Yeah, it is a very nice question, but also very challenging. Yeah, I know. Yeah, I know Miss Umi. Yeah. She is just testing me. Yeah? So yes, we have to have, yeah, by any means possible, we have to have this like NCBI like data. This is actually one of the purpose of INPEL. Yeah, so although INPEL later on will be a very small part of NCBI, for example, because there will be a genomic, for example, at ECMN. Yeah, ECMN have a genomic national center, but it's actually not national. This is the term national here. We are still confusing. The term national, it means it should be open for all universities, should be open for all institutions, not only for its own institutions. Yeah, the national centers should organize all these activities the same. For example, National Genomic Center, Malaysia have Malaysia Genomic Center. We don't have Indonesia Genomic Center. But once we have this Indonesia Genomic Center, then it will coordinate, yeah, all these genomic activities, not only at that particular institution, but also in all universities and also research institutions. Anything related to genomic research, then it has to be under coordination of the National Genomic Center. Of course, this also applies to any other national centers. Yeah, in the US, for example, it's very common. We have, for example, in my university, National Asphalt Center. Everything related to asphalt, they will refer to this university. And other Los Alamos National Centers, everything research related to this, it will coordinate with this. The answer is yes. Yeah. Okay. But we don't know how long. Okay.
Okay, I think this is very big question to us, yeah. And then we looking forward that NCBI Indonesian version will be available soon. This is the last question, last but not least, is Prof. Kholis, what about the plants that included in plant conservation group? Is it still accessible to extract plants active compounds from this kind of plants?
Yes, very good question. The idea if extraction plant is not that we are going to harvest all the plants. Okay, please keep this in mind. The idea of utilizing plants is just to find the compounds. First, to identify the compounds. If it is the ordinary compounds, then that's good. We don't have to synthesize the compounds; it's already there. But if, let's say, we identify new type of compounds and we do not find this, then we can do synthetic biology, right? We can synthesize this. Of course, it's not always easy, but for example, mangrove. Actually, we can plant mangrove. We can grow mangrove, and it will be very good for society, for communities. If, let's say, we have a very potential mangrove, yeah, as I already found several ones, that can be growth, yeah, in rural areas, in long of Indonesian coastline. Remember, Indonesia has around 90,000 kilometer coastline, so you can pick any place that can grow our mangrove, different type of mangrove. Okay, thank you.
Okay, thank you very much, Dr. Kholis. And I think this is the end of the session of Keynote. And thank you very much for all speakers, from Professor Ahmad, Professor Fahrur, from Mrs. Tan, and Dr. Kholis for a very interesting talk from the morning until now. And then I noticed that the participant is still exists, yeah, still keep patient and follow all the lecturer from the experts. And me, Komeni Hassan, as a moderator, I'm apologize for all weaknesses and mistake to all of you. And I would like to thank you to all of you to keep it stay in this session. And I will give it back to the organizer. And thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for many.
Ladies and gentlemen, before we end this plenary session, we have to take a photo. Please, switch on your camera. Okay. I will count to 1, 2, 3. 1, 2, 3. Okay. Once more. 1, 2, 3. Okay. One more time. 1, 2, 3. Okay, once more. 1, 2, 3. And the last. 1, 2, 3.
Thank you all very much.
Yes, thank you for the participant. Now, before we were going to the parallelization, I will give you a quick demonstrate how to go to your room, to go your parallelization room. Okay, you will to fill this registration ID first, like this, and you click login, and then you have to go in conference room, and you click the menu parallel session. Sorry. And then you will find your name. For the example, Badru Tamam, you have to access room 1. Is that clear, everyone? For the instruction? Hello?
Yes, it's clear.
It's clear. Okay. Okay. If without any further ado, you can go to your parallel session room. Thank you for your attention, and thank you for the moderator, Dr. Khomeini, and the keynote speaker, for the all keynote speaker. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much. Yeah.
For the all participant, you can leave this main room. You can go to your parallel session room.